Hello there, and welcome to Life Beyond Seen, where curiosity gets comfortable and science is allowed to slow down for a while. Tonight, we're heading to a place few people will ever see with their own eyes. A place so deep, so cold, and so far from daylight that even the idea of a skeleton on the seafloor sounds a little unreal at first. And yet, down there, when a whale dies and its body sinks through the water column, the story is not over. In many ways, it is just getting started. Because on the ocean floor, bones do not always sit quietly and wait to be covered by mud. Sometimes they become food, not for sharks, not for crabs with great crushing claws, not for some giant deep-sea monster from an old sailor's nightmare, but for worms. Small, strange worms. Worms that do not look built for chewing. Worms that do not even have mouths. Worms that settle into bone and begin, very patiently, to make a meal out of it. That is our subject tonight. The worms that eat bones on the ocean floor, known as Osedax. The name means bone devourer, which is a very direct bit of naming and honestly saves everyone time. Scientists first described Osedax from whale bones in Monterey Canyon after a whale carcass discovered there in 2002 revealed animals unlike any that had been known before. They turned out to be annelid worms, related to cibberglinid tube worms, and the first described species included Osedax rubiplumus and Osedax frankpressi. Before we get into any of that, I'd love for you to take a quiet second and notice where you are listening from tonight. Maybe you're in bed already, maybe you're on a sofa with one lamp on, maybe you are winding down after a long day and pretending you will only rest your eyes for a moment, which history tells us can be a very optimistic plan. And what time is it for you right now, in your part of the world, while we talk about a place where clocks mean nothing and the nearest sign of morning is hundreds or even thousands of meters above? If this sort of slow scientific wandering is your kind of evening company, and if you find it soothing or just pleasantly odd in the best possible way, I'd be very grateful if you gave the video a like and maybe subscribed as well. It helps this small quiet corner reach more people who enjoy drifting off with something curious to think about. And now, let's settle in and take this one piece at a time, because Osedax is one of those animals that sounds invented until you start looking at the details and then somehow becomes even stranger. To start in the most ordinary place possible, think about a bone. Not as a symbol, not as something from a museum drawer, but as actual material. Hard, dense, pale, dry-looking, at least from the outside. It does not seem like an easy meal. If someone told you that an animal lived on bone alone, you might reasonably picture powerful jaws, sharp teeth, crushing force, or at the very least some determined nibbling. You would not picture a soft-bodied worm with feathery plumes. You would certainly not picture a worm with no mouth, no eyes, and no stomach. Yet Osedax females have exactly that kind of body plan. They show red or pinkish palps and a trunk above the bone, while a root-like structure penetrates the skeleton below, and the animals depend on bacteria associated with those tissues to help unlock nutrition from the bone. That alone would be enough to make them memorable, but they are not simply odd for the sake of being odd. They make sense in the setting where they live. The deep sea is not a place where food drifts by in generous portions every afternoon. A large carcass falling to the seafloor can become an important source of energy in an otherwise food-limited environment, and whale bones in particular can persist long enough to support a whole sequence of scavengers and colonists. MBARI researchers have described whalefalls as feasts in nutrient-poor surroundings, and Osedax became especially important because they consume the bones, which hold a major remaining store of energy once the soft tissue is gone. There is something almost practical about the whole arrangement. On land, when a large animal dies, the cleanup crew arrives quickly and in force. Flesh is stripped, bones are cracked, marrow is reached, nutrients are scattered and recycled. In the deep ocean, the cast is different and the pace is slower, but the same broad rule still applies. Nothing useful is left untouched for long. First come the large scavengers for the soft tissue. Later, when the skeleton remains, Osedax can move in and deal with what is left. They do not rush, they do not tear, they settle, grow, extend into the bone, and stay with the job. Whalefall studies in Monterey Bay showed that after larger scavengers removed flesh, Osedax colonized the remaining bones at multiple depths. This is part of what makes them such a good subject for a slow nighttime story. So much of nature that gets attention is loud, bright, fast, or visibly dramatic. We notice the pounce, the chase, the splash, the strike. Osedax does none of that. It lives in darkness. It works by contact and chemistry. It makes its living inside remains that most other animals would pass over once the easy parts were gone. It is almost the opposite of showy. And still, once scientists found it, they realised they were looking at something important. These worms were not just another strange detail in the deep sea. They were helping decide what happened to big vertebrate skeletons after death, and that means they were helping shape how nutrients move through those seafloor communities. Embry researchers later described Osedax as a foundation species at whale falls in Monterey Canyon because of how strongly they influence the fate of the bones, and in turn, the community that depends on them. I think that is one reason people respond so strongly to them. On first hearing, bone-eating worm sounds like one of those phrases built to make you sit up a little straighter. It has the right amount of menace, even though the animal itself is small and not remotely interested in you. It sounds like something from a horror film trailer, right up until you see the actual creature and realise it looks more like a tiny tufted flower that made some very unusual life decisions. The exposed parts can be delicate and colourful. The hidden parts do the serious work. In that way, Osedax is a useful reminder that nature is under no obligation to match our expectations. If there is a resource somewhere, some organism will eventually find a way to use it. In this case, the resource is locked inside a structure most animals cannot easily exploit. Bone is rich, but it is not open for business. To get at it, Osedax uses a combination of anatomy and chemistry that researchers have spent years trying to understand. Studies on Osedax roots found evidence of strong acid-secreting ability and enzymes linked to dissolving the mineral part of bone, helping expose the organic material within. Other work has highlighted the role of symbiotic bacteria and associated microbes in breaking down collagen, cholesterol, and lipids from the skeleton. So when we say these worms eat bone, it is not a matter of tiny mouths taking neat bites. It is a much stranger system than that. And already, without getting too far ahead, you can see why the opening image matters so much. A whale falls, scavengers arrive, time passes, the bones remain. Then, eventually, from the water above or nearby, larvae settle. A few thread-like bodies begin to establish themselves. Little plumes appear. Tubes form. Roots spread into the skeleton. What looked like the final leftover structure starts turning into habitat and food at the same time. It is one of those deep-sea stories that makes death look less like a sudden stop and more like a transfer of materials from one set of living things to another. Nothing sentimental about it. Nothing neat. Just biology being very thorough. There is also a good chance that Osedax has been doing versions of this far longer than whales themselves have existed in anything like their modern form. Early ideas linked the worms closely to whale bones, which made sense at first, because that is where they were discovered. But later work showed they can colonize other bones as well, including cow bones placed experimentally on the seafloor, and MBARI researchers reported that they fed on many kinds of bones in artificial carcass experiments. Fossil traces attributed to Osedax-like activity have even been reported from much older marine vertebrate remains, suggesting the lineage may predate whales and once exploited bones from marine reptiles and fishes. That changes the picture in an important way. It means we are not simply looking at a worm that evolved to take advantage of modern whales. We may be looking at an older strategy, one that found a way to use dead vertebrate skeletons in the sea whenever and wherever such opportunities appeared. Whales are just the large, long-lasting, easy-to-find examples that helped scientists notice what was happening. A tuna vertebra on the seafloor may disappear too quickly, or be too hard to detect for us to catch the same process in the act. while a whale skeleton stays put long enough to become a kind of public notice board for anyone in the neighbourhood with the right set of adaptations. MBARI researchers made exactly this point when discussing why whale bones are so often where we find the worms. Of course, that only makes the actual discovery more enjoyable. Imagine being part of a team studying a dead whale in deep water. You expect scavengers, you expect decomposition, you expect perhaps a sequence of known animals doing familiar things. Then you notice patches on the bones. You investigate more closely. The worms are dense on the skeleton. They do not match anything already described. Their structure is unusual. Their way of feeding is stranger still. And then, as if the story needed one more twist, the adults you're looking at turn out mostly to be females, and the males are tiny, larval-looking dwarves living inside the females' tubes. The original work on Osedax highlighted these numerous dwarf males, and later studies showed that some females can accumulate many males, creating a dramatically biased sex ratio. That is the sort of detail that would sound excessive if a novelist used it too early. You would tell them to edit it back a little. Keep the worm that eats bone if you must, but perhaps let it have a normal family structure. Nature, as usual, declined that suggestion. Still, I do not want to rush ahead into the sex lives and settlement patterns and root chemistry before we have properly arrived at the setting, because setting matters here. Osedax is not strange in a vacuum. It is strange in a specific place, under specific conditions, on a specific food source that appears only occasionally and then changes over time. A whale fall is not just a dead whale on the seabed, it is an event. It alters the local environment. It attracts a succession of animals and microbes. It creates a temporary island of abundance in a vast area where meals are usually small and scattered. By the time Osedax appears, the easy feeding stage is mostly over, and the bones are becoming the main storehouse that remains. And there is something useful, I think, in picturing that patiently, not in a dramatic cinematic way, more like a long-term process on the ocean floor. A huge body descends, it lands. The first consumers arrive, tissue is removed, sediment shifts around the carcass, other animals gather, bacteria respond. The skeleton changes from support structure to food reserve. Then these worms, delicate above and invasive below, start converting dense hard remains into something the surrounding ecosystem can use. Carbon that might otherwise stay locked away longer in a buried or slowly decaying skeleton gets moved through the food web and into nearby sediments. That broader role in carbon cycling has been emphasised by MBARI's reporting on Osedax and whalefall research. When you hear that, the animal starts to look a little different. Less like a deep-sea oddity collected for a list of bizarre creatures, and more like a worker in a system. A specialist, yes, but also part of a chain of transfer. It is taking something stubborn and making it available. Bone is not the end of the story. Bone is stored energy behind a locked door. Osedax comes with the key, or at least with the biological equivalent of a very determined locksmith. The funny part is that if you met one without context, you might not guess any of this. You might see a plume and think of a filter feeder. You might see a tube and assume a quiet, ordinary worm life. You would not immediately infer acid secretion, symbiotic microbes, buried roots, dwarf males, and a career centred on whale remains. This is one of the pleasures of marine biology, especially in the deep sea. The body plan is often only the first clue. The lifestyle is where things get truly unreasonable. Even their relatives help make the point. Osedax belongs within the cyberglinids, the group that also includes famous tube worms from hydrothermal vents and cold seeps, animals already known for unusual symbioses and reduced digestive systems. The link was recognised quickly after discovery through DNA evidence and morphology. So in one sense, Osedax was startlingly new, and in another sense it fit into a broader pattern the deep sea had been hinting at for some time. If an environment offers an energy source that is hard for most animals to use directly, there is a decent chance some worm-like specialist has quietly solved the problem already. There is another reason this topic works well at night. It forces the mind to slow down and accept timescales that are not built around our own routines. Most of us live by hours, deadlines, commutes, meal times, and those strangely fast 10 minutes before we're supposed to leave the house. Down on the seafloor, the schedule is different. A carcass may take time to descend. Scavenging happens in stages, colonization happens in stages, growth happens in stages, even discovery happens in stages, with researchers returning to bones again and again, watching communities change. MBARI's whalefall work involved repeated visits to carcasses placed at different depths, following the decomposition and colonization process over time. That slower frame changes the story. Osedax is not a sudden attacker, it is a patient arrival. It waits for the right resource, it settles where conditions allow. It develops in contact with a food source that is both durable and limited. In some places and conditions, these worms may flourish. In others, they may fail to colonize or arrive late. Recent and older field studies alike show that whalefall communities vary with conditions such as oxygen and site characteristics, and Osedax presence is not identical everywhere. So tonight, as we begin properly, that is where I want to leave the camera for a while. Not on the whole life history yet. Not on the full chemistry. Not on the remarkable males tucked away in female tubes, or on the later questions about fossils and evolution, or on the experiments with non-whale bones, though we will get to those. For now, I just want the scene in place. Far below the surface, a skeleton lies on the seabed. The flesh that once powered one of the largest animals on Earth has mostly gone. The bones remain, rich and difficult, dense with material most creatures cannot reach. Around them, the deep ocean carries on, cold and dark and not especially concerned with whether anything up above knows this is happening. Then, in time, small worms arrive. They do not come with teeth. They do not come with speed. They come with plumes, tubes, roots, bacteria, and a solution to a very specific problem. And once you know that, the whole seafloor starts to look a little less empty than it did a few minutes ago, which is exactly where we need to be before we move closer and look at what kind of place could support an animal like this in the first place. Far below the surface, much farther down than sunlight can manage, the seafloor is usually a place of limited meals and long waiting. Most of the food that reaches those depths comes as a slow drift of small particles from above, bits of dead plankton, fecal pellets, shredded remains, and other organic debris that marine biologists often call marine snow. That steady fall matters enormously, but it is thinly spread, and in many deep places animals live on modest rations for very long periods. Whalefalls stand out because they interrupt that scarcity with a huge delivery of organic material all at once. Review work on whalefall ecosystems describes the deep sea as energy poor compared with the surface, notes the importance of marine snow to most deep habitats, and explains that a whale carcass can act like an oasis or habitat island on the seabed. That shift in scale is worth pausing on, because it changes the whole mood of the environment around the carcass. A whale that reaches the seafloor is not just another dead animal in a place full of leftovers. It is a very large pulse of energy entering a habitat where food often arrives in crumbs. One review cited in Frontiers notes that the soft tissue of a 30-ton whale contains about 1.2 times 10 to the third power kilograms of organic carbon, roughly comparable to the background particular organic carbon flux to 100 square meters of deep sea floor over 1,000 years. The same review adds that whalefalls can deliver organic matter far faster than the normal downward rain of marine particles. So when a carcass lands, the surrounding seabed is not seeing a slightly better lunch than usual. It is seeing an event big enough to reshape local chemistry, local traffic, and local survival prospects for a long stretch of time. The first effect is simple and unsentimental. Animals that can smell or detect the carcass arrive and begin feeding. In shallower or more accessible settings, that often means sharks, hagfish, large crabs, fish, and other mobile scavengers. At some great depths, and in some locations the cast can be different, but the basic pattern remains. The easy food goes first. In work from Monterey Canyon, researchers described a mobile scavenger stage that can remove more than 90% of the soft tissue, often within about 4 months after a carcass reaches the seafloor, depending on its size and circumstances. Embere's later whale carcass studies describe these carcasses as feasts in nutrient-poor surroundings, revisited over time as different groups of consumers took over. So the quiet feast is not quiet because nothing happens. It is quiet because it happens where no human ear can hear it, and because the pace is measured in visits, not in sudden spectacle. Even in that first stage, the whale is already doing more than feeding the obvious guests. Tissue falls into sediment, fluids leak out, microbes respond. Nearby animals that are not eating the carcass directly can still benefit from the enriched patch around it. That broader effect is one reason whale falls are treated as ecosystems rather than single objects. The Monterey Canyon study described changes in surrounding sediments, microbial mats, and polychaete composition over repeated visits, while broader reviews emphasize that large carcasses enrich the seafloor locally and can influence abundance and diversity well beyond the skeleton itself. In other words, a whale fall does not merely host consumers on its surface, it changes the patch of seafloor around it into a richer place than it was the week before the carcass arrived, and that difference can persist long after the obvious flesh is gone. After the large scavengers have taken the easy parts, the story does not end or even really slow down. It changes character. Researchers studying whale fall succession describe an enrichment opportunist stage, often dominated by smaller invertebrates such as polychaetes and crustaceans that thrive in the organic rich sediments and leftover tissues around the remains. In the Monterey Canyon framework, that stage may last up to about 2 years. This is part of what makes whale falls so useful for thinking about deep-sea life in general. They do not support one burst of activity followed by emptiness. They support a sequence. Each phase opens opportunities for a different set of organisms, and each group alters conditions for what comes next. A carcass becomes less like a single meal and more like a changing neighbourhood, with one tenant moving in as another leaves, and with the building itself slowly being taken apart while everyone is still using it. By the time that process reaches the skeleton, the whale has become something more specialised. Bone is harder to exploit than flesh, but it is still rich. Great whales carry substantial lipids in their bones, and as those lipids break down, they can help support microbial processes that generate sulfide-rich conditions. Reviews of whale fall ecosystems emphasize that the high bone lipid content of large whales is one reason the carcasses can support a sequence of heterotrophic and chemosynthetic communities in the deep sea. The Monterey Canyon study described a later sulfophilic stage that can last for years or even decades depending on carcass size, and can support microbial mats plus a large diversity of invertebrates adapted to sulfide-rich settings. So the skeleton is not merely what remains when the real food is gone. It is another reservoir, locked up differently, and one that can keep feeding life long after the body has stopped looking like a body at all. This helps explain why whale falls are often compared with the islands. Out across a vast seafloor where resources are usually sparse and patchy, a large carcass creates a concentrated temporary habitat with its own chemistry and its own community. Some papers and news coverage use the word oasis, and that is not an exaggeration when speaking ecologically. The carcass offers food, shelter, microbial activity, and in later stages even conditions that resemble other reducing environments such as vents or seeps in some limited ways. The annual review abstract describes whale falls as organic and sulfide-rich habitat islands. The 2004 Monterey Canyon paper likewise discusses them as habitat islands that may occur along whale migration corridors and influence abundance and diversity in deep regions. That patchiness matters because deep-sea specialists may need such sites not just to feed, but also to disperse from one suitable patch to the next over generations. Seen from that angle, the word feast starts to mean something a little broader than eating. It is not simply that many mouths can gather at one place, it is that whole systems get to do more than usual for a while. Microbes multiply, scavengers breed or grow, opportunists colonize, sulfide-tolerant animals arrive, specialists such as Osedax settle in when the conditions suit them. A Frontiers Review reports species richness at whale falls, reaching hundreds of species across studies, and notes their importance to deep-sea biodiversity and carbon transfer. Even where precise totals vary from site to site, the larger point is consistent across the literature. One carcass can support far more biological activity than the same patch of bare, deep sediment ordinarily would. So the feast is quiet, but it is crowded in its own way, and crowded by the standards of a place where many animals usually spend much of their time waiting for whatever drifts down next. It is also important not to imagine every whale fall as identical. Depth matters, oxygen matters, location matters, the pool of nearby colonizers matters. In the Monterey Canyon study, researchers were careful to note that succession is shaped not only by facilitation between stages, but also by dispersal abilities, bathymetric ranges, and chance. Their deep carcass, at 2,891 metres, gave them an unusual opportunity, because some larger mobile scavengers that are common at shallower whale falls were less active or absent at that depth. Embari's later experiments deliberately placed whale carcasses from 385 to 1,820 metres and revisited them over time, comparing how different conditions influence colonisation by Osedax and other fauna. So while the broad sequence is useful, the details can vary a great deal. A whale fall is a process with common themes, not a rigid script followed exactly the same way every time. That variability makes the arrival of Osedax even more interesting. These worms are not early loud participants in the cleanup. They are specialists tied most strongly to the phase when bones have become the main remaining store of nutrition. MBRE researchers observed Osedax on remaining bones at all the experimental depths once larger scavengers had stripped the carcasses. Later, MBARI reporting described the worms as largely controlling the fate of whale falls in Monterey Canyon because they consume the primary remaining nutritional source, the bones, and therefore influence how long a whale fall community can persist. That is a remarkable role for an animal that looks, at first glance, like a small tuft, rather than a major ecological force. It means the end of the feast, in one sense, depends on when and how effectively these worms turn the skeleton itself into food and habitat for others. There is a nice practical logic to that. A whale skeleton is durable. It can remain in place and hold energy for years. But it does not become broadly available unless something can open it up. Osedax is one of the clearest answers nature has produced to that problem. Smithsonian Ocean explains that these worms do not chew mineralized bone directly with mouths because they have no mouths or stomachs. Instead, they dissolve bone material and rely on symbiotic bacteria in the process of obtaining nutrients from fats and proteins within the skeleton. That feeding style fits neatly into the whale fall story. Once the flesh is gone and the bones stand out as a stubborn storehouse, Osedax arrives with a method suited exactly to that hidden reserve. The feast by then is no longer about tearing pieces off a carcass. It is about unlocking a sealed pantry. If this all sounds oddly efficient, that is because it is. The deep sea tends to reward anything that can make use of resources others leave behind. Large carcasses are too valuable to waste, and a whale offers several rounds of opportunity in one body. Flesh goes first. Enriched sediment feeds opportunists next. Bone lipids and associated microbial processes sustain later communities. Specialists then make use of materials that would otherwise remain difficult to access. This layered use of a carcass is one reason whale falls have attracted so much attention from ecologists. They reveal, in a form that is unusually easy to notice, how deep sea life is organised around rare pulses of food and around organisms capable of exploiting different stages of decay. They also show how one dead animal can keep altering its surroundings for years. There's another side to the story that makes the whole scene even more striking. Whale falls do not just matter locally in a single moment, because they may occur along migration routes and in regions where many large whales live or once lived. They could form a scattered network of stepping stone habitats through deep basins. The Monterey Canyon paper notes estimates that hundreds of grey whales may sink annually in a large area of the Northeast Pacific, and later reviews discuss whale falls as important stepping stones for dispersal and biodiversity in the deep sea. That does not mean every square kilometre is dotted with skeletons waiting to be found. Far from it. But it does mean the deep seabed is not as disconnected as it appears if one only imagines bare mud between vents and seeps. Carcasses may link those spaces ecologically across long periods, especially for organisms whose larvae can travel, and whose adults can exploit bones when they find them. That network idea also gives the quiet feast a slightly different scale. It is one thing to picture a single skeleton being consumed in darkness. It is another to realise that over evolutionary time, repeated carcasses may have provided recurring opportunities across ocean basins, supporting specialists and perhaps helping some lineages spread or diversify. The annual review abstract specifically notes that whale falls have yielded insights into ecology, paleoecology and evolution, and that they may have served as hotspots of adaptive radiation for specialised fauna. Osedax belongs squarely in that larger story. It is not just feeding on a single dead whale in one canyon. It is part of a way of life built around rare, rich islands of bone on the seafloor, repeated often enough over vast timescales for specialisation to become worthwhile. All of this helps explain why researchers were so excited when deep whale carcasses in Monterey Canyon and elsewhere were revisited again and again. Those sites let scientists watch succession in a setting where the rules of deep-sea feeding become unusually visible. Instead of inferring everything from sparse remains or sediment chemistry alone, they could observe who arrived, when they arrived, what disappeared, what lingered, and how the community changed. The 2004 Monterey Canyon study reported visits at 0, 1, and 8 months after discovery, documenting tissue loss, microbial mat development, and changes in fauna. MBARI's later deployments extended that approach across multiple carcasses and depths. A whale fall, in that sense, became both an ecological event and a natural experiment, one that let the deep sea reveal itself in stages. And perhaps that is the best way to hold this part of the story in mind before moving any closer. The seafloor is not empty, but it often runs on very small budgets. A whale fall changes that. It creates a local abundance so large that it can feed scavengers, enrich sediment, support microbial chemistry, sustain specialised invertebrates, and keep doing useful ecological work for years. What begins as a carcass becomes a crowded process. What looks, from a distance, like the end of one life becomes a long set of openings for many others. Osedax belongs to the later part of that process, when the skeleton has become the main prize and the feast has shifted from flesh to bone, which is exactly where we need to look next if we want to understand how scientists first realised that a quiet patch of whale skeleton on the ocean floor was home to something entirely new. Once a whale fall has turned the seafloor around it into a place of unusual traffic, the next part of the story depends on someone noticing that not everything on the bones belongs there in any familiar way. That is exactly what happened in Monterey Canyon in 2002, when a research team from MBARI was not out looking for bone-eating worms at all. Bob Vrian Hook and his colleagues were on a cruise searching for clams, following a large sonar signal on the seafloor with the remotely operated vehicle Tiburon. What they expected was one kind of deep-sea community. What they found instead was a sunken grey whale carcass, and spread across the bones was what MBARI later described as a shaggy red carpet of unusual worms. The carcass lay at about 2,891 metres depth in the axis of Monterey Canyon, around 31 kilometres offshore, and it represented a major pulse of organic material in a food-limited setting. That matters because discoveries in biology are often less theatrical than people imagine. There is no dramatic music, no one gasps and drops a coffee mug in slow motion, and the animal itself has no sense that it has just altered someone's career plans. There is usually a screen, a feed from a camera, a cluster of people trying to make sense of what they're seeing, and then a practical question. What exactly is that? In this case, the answer was not obvious. The worms carpeting the skeleton were not merely another expected scavenger. They looked unusual enough that the researchers quickly realised they might be looking at something undescribed. MBARI's account of the discovery makes the sequence very clear. The team watched Tiburon approach the target, expected clams, and instead found a dead whale with bones covered in bizarre red worms. There is something especially satisfying about that kind of discovery, because it comes from paying attention rather than chasing a legend. Nobody was sent out with a mission called Find the Bone Worms. They were doing ordinary scientific work in an extraordinary environment, and the deep sea offered them a surprise. That surprise was not subtle. In photographs from the site, the bones really do look furred or tufted with life. The visible parts of the worms were reddish plumes and trunks, the sort of structures that, from a distance, could almost make the bones look overgrown rather than inhabited. MBARI's 2004 release notes close-up views of whale bones covered with Osedax at almost 3 kilometers depth, while the 2017 annual report recalls the red patches on the bones as thousands of deep-sea worms. At that point, however, seeing the worms was only the beginning. The real work started when the team collected specimens and tried to place them in the tree of life. And here the story becomes very recognisably scientific in the best way. First comes the visual oddity, then come the samples, then comes the puzzle of anatomy, genetics, comparison, and a growing suspicion that the animal in hand may not fit any available label. MBARI reports that within 2 days of the discovery, DNA sequences had already shown that the worms were related to deep-sea tube worms from hydrothermal vents and cold seeps. That was a major clue, but not a complete answer, because related does not mean identical, and these worms had a body plan that was strange enough to leave even experts uncertain at first about what they were looking at. Greg Rouse, then at the South Australian Museum, was brought in to help identify them, and after closer examination he confirmed that the worms were annelids and relatives of deep-sea tube worms, but entirely new to science. That quick DNA step is worth lingering on for a moment, because it captures how discovery really works now in many areas of biology. 100 years earlier, the entire classification might have depended far more heavily on visible anatomy from the start, which would have made this case even more awkward. Osedax females do not make identification easy. They have no eyes, no legs, no mouth, and no stomach. They have feathery plumes exposed to the seawater, a muscular trunk that can withdraw into a tube, and below that a hidden structure including an oversac and branching roots embedded in the bone. Those roots house bacteria and are tied to the worm's feeding strategy. That is not the sort of body most biologists carry around as a standard reference image in their heads. MBARI's descriptions from 2004 and 2017 both emphasise how unusual the anatomy appeared at first sight. If anything, the worms became stranger under the microscope rather than simpler. The initial collections appeared to consist only of females. That alone would make any careful researcher slow down and check again. Either the team had sampled in an oddly biased way, or something much less ordinary was going on. It turned out to be the second option. While examining females that range from about 2 to 7 centimetres long, researchers found tiny males associated with them, microscopic worms that looked as though they had never developed past a larval stage, and that contained yolk remnants along with large quantities of sperm, MBAR. Some accounts explain that these dwarf males live within the gelatinous tube surrounding each female, and can occur in numbers ranging from several to hundreds per female. This was not just a new species, it was a new genus with a reproductive system dramatic enough to make the rest of the anatomy look almost restrained by comparison. And this is where the idea of a garden in the skeleton starts to make practical sense. A garden is not simply a place where living things are present. It is a place where many individuals have established themselves densely on a substrate, where growth is concentrated, and where what looked like bare structure has turned into a living surface. That is what the whale bones had become. They were not just leftovers from a carcass. They were colonized space, crowded with plumes above and roots below. The bones supported a settlement dense enough to read visually as coverage rather than scattered occupancy, and the deeper researchers looked, the more layered that community became. The Monterey Canyon whale fall was already known to host unusual benthic fauna, including new polychaetes, microbial mats, and other opportunists, but Osedax gave the skeleton a more intimate kind of colonization. These worms were not merely sitting on bone, they were entering it. By 2004, the team had gone far enough to publish the formal description in Science, naming two species, Osedax rubiplumus and Osedax frankprisi, from that Monterey Canyon grey whale carcass. MBA-Eyes release identifies the paper directly as Osedax, bone-eating marine worms with dwarf males, and notes that the two named species were described from the whale fall first seen in 2002. The new genus name, Osedax, was chosen from Latin for bone devourer, which is admirably straightforward and spares future students from having to guess whether the animal was named after a Roman official, a ship, or someone's uncle. It tells you the job right there in the title. Still, a formal name only settles one part of the mystery. The next problem was understanding how the animal actually lived. A worm without a mouth and without a stomach has forced its discoverers into a very specific line of inquiry. If it is thriving on whale bones, how is that possible in mechanical terms? Here again, the hidden structures mattered more than the visible ones. MBARI reported that the greenish roots branching from the lower body were filled with bacteria that break down oil in the whale bones, and later descriptions emphasized that the worms burrowed into bone and used symbiotic bacteria to help extract fats and oils. More recent work has expanded that picture with evidence for acidification and enzymatic digestion associated with the root tissues, but the key point was already visible in the first years after discovery. The worms were not nibbling bone surfaces in any conventional sense. They had evolved a completely different route into the resource. That combination of visible plume and hidden root probably helped the worms evade notice before then, not because they were impossible to see, but because the right circumstances had to line up. Researchers had to find an appropriate carcass, at the right stage, at great depth, revisit it, and take a close enough look to recognise that these were not just ordinary occupants of a whale fall. Even then, it helped enormously that MBARI had the equipment and flexibility to follow up. The Institute's later reports stress how well placed it was to continue the work. Dead whales that washed ashore between 2004 and 2008 could be recovered, weighted, towed offshore, and deliberately sunk at different depths in Monterey Canyon, after which the carcasses could be revisited with ROVs at multiple stages of decay. That experimental follow-up transformed a striking accident into a sustained research program. Those later deployments also taught researchers something important about the original discovery. At first, because Osedax had been found on whale bones, it was tempting to think of the worms as specialists on whales alone. But that idea ran into trouble quickly, both from genetic clues and from experiments. MBARI reports that initial molecular analyses showed unexpectedly high genetic diversity in Monterey Bay Osedax, too much for a lineage supposedly living only on rare modern whale skeletons. That led Frey and Hook and colleagues to test other bone types by placing cow, tuna, turtle and turkey bones on the seafloor. The worms colonised many kinds of bone. In other words, the first discovery happened on a whale because whales provide large, persistent, easy-to-find bone habitat, not because Osedax was necessarily limited to whales. That realisation widened the implications of the original find considerably. There is a pleasing bit of scientific humility in that sequence. First, the animal seems tied to one spectacular setting, then the data suggest otherwise, then experiments test the idea. then the picture changes. It's a good reminder that the first version of a discovery story is rarely the final one. The initial sighting in Monterey Canyon gave the world the animal, but not the full scope of its habits. Only with more carcasses, more samples, and more years of patient work did researchers start to see how broadly Osedax could use vertebrate bone and how diverse the genus really was. A 2009 review described Osedax diversity as remarkable, noting that the genus, first described from Monterey Bay whale bones recovered from around 2,893 metres, rapidly expanded to include species from multiple depths and oceans. MBAR later reported that dozens of species had been observed worldwide. Yet the original Monterey Canyon skeleton retains a certain importance because it gave the first clear view of the whole package. The worms were abundant enough to notice. Their anatomy was strange enough to demand explanation. Their relationship to cypoglinid tube worms linked them to a broader deep-sea story. Their dwarf males made them unforgettable. Their feeding methods suggested a completely new way of exploiting marine vertebrate remains, and their presence on a whale fall connected them to an ecosystem already known to proceed in stages, from scavengers to opportunists to sulfide-based communities. In a single site, researchers got not just a new animal, but a new ecological role to think about. There is also the matter of timing, which often gets missed because the animal itself is so eye-catching. The discovery came during a period when deep-sea exploration technology was making it increasingly possible to revisit precise sites, recover delicate specimens, and combine morphology with molecular work quickly. MBARI's use of Tiburon, along with repeated dives and later controlled carcass deployments, allowed the team to move from "this is odd" to "this is a new genus with this anatomy, these relatives, and these likely ecological functions," in a way that would have been much harder in earlier eras. The whale fall itself was still doing what whale falls have always done. What changed was our ability to watch closely enough and long enough. That may be the most satisfying part of the story. A deep-sea whale skeleton, from a human point of view, looks like the sort of place where almost nothing ought to be left to surprise us. It feels like the late stage of a process, nearly the end of the usable material, the stripped framework after the interesting part is over. But scientists looked at those bones and found not emptiness, not finality, and not merely a few leftovers, but a densely occupied surface and a feeding strategy nobody had documented before. They found a red living layer on white bone. They found roots where they had expected perhaps only tubes. They found a new genus inside a carcass that had already been partly consumed. In that very literal sense, they found growth in the skeleton, a living population established where many people would have expected mostly silence. Once that happened, the bones of dead whales stopped being just one stage in decomposition and became something else as well. A site of discovery, a repeated experiment, and a clue to a deeper network of specialists moving through the deep sea from one hard meal to the next. The first sighting in 2002 opened a line of research that spread quickly to Sweden, Japan, and beyond, where intentionally sunk carcasses also drew Osedax within a year, confirming that Monterey Canyon had not produced a one-off curiosity. It had revealed a real lineage, already out there and already widespread, simply waiting for someone to notice the garden on the bones. And once the existence of that garden was clear, the next step was unavoidable. It was no longer enough to say that strange worms lived on whale skeletons. Scientists now had to explain what kind of animal could live that way at all, which brings us directly to the body itself and to one of the oddest designs the deep sea has produced. A worm that manages to eat bone without the usual equipment most of us would consider basic for eating anything. What scientists had found on those whale bones was not just a new species with a strange diet. It was a body plan that seemed to ignore several pieces of equipment most animals would consider basic. Osedax females have no eyes, no legs, no mouth, and no stomach. Yet they are perfectly capable of settling on a bone, growing there, reproducing there, and drawing nutrition from one of the least inviting meals in the ocean. MBAR's summary of the group puts it bluntly. These worms lack mouths and guts, but they do have colourful plumes and a root system that digs into bone. Smithsonian Ocean says much the same, noting that Osedax digests nutrients from bone without a mouth or stomach at all. That sounds impossible only if one imagines feeding as a matter of chewing, swallowing and digesting in the familiar way. Osedax has solved the same problem from a different angle. Instead of taking food into a digestive tract, the female worm stays anchored to the bone and uses her lower body to get into the skeleton itself. The visible part above the bone looks almost tidy by comparison. Emerging from the bone is a cylindrical trunk, usually enclosed in a transparent or gelatinous tube, and at the top there is typically a crown of feathery palps. A neural reconstruction study summarised in search results describes this basic arrangement clearly. A trunk rises from the bone, enclosed in a transparent tube, and generally bears a plume of 4 palps. Those palps are the part most likely to mislead the eye. They give Osedax a soft, almost plant-like look, as though the worm was something decorative growing from a crack in the skeleton. In living animals, the palps are often red or pink because they are rich in blood, and they function largely in gas exchange, drawing oxygen from the surrounding seawater. Smithsonian Ocean notes that the feathery plumes act as gills and can be retracted when the animal is disturbed. Work on the respiratory system of Osedax mucofloris also showed that the palps are built for efficient oxygen uptake, which makes sense for an animal whose hidden tissues and microbial partners need a steady supply of oxygen to keep their chemistry running. So the upper half of the worm is doing one kind of work, and the lower half is doing something much stranger. Below the trunk, inside the cavity the worm is established in the bone, lie the ovisac and the branching root system. A Royal Society summary of the acid secretion study notes that the female lower trunk, ovisac, and roots all lie in a cavity excavated in the bone, and that the roots may branch to create an extensive network. That detail matters because it shows just how misleading the external view can be. What looks above the surface like one small worm is, in functional terms, only the visible top of a larger structure. Much of the important business is happening below the bone surface, out of sight, in a place the worm has chemically opened for itself. This is where the phrase "no stomach" stops sounding like a deficiency and starts sounding like a design choice. Osedax does not need a conventional gut because it is not processing chunks of food internally in the usual way. The lower tissues and their microbial partners do the first heavy work at the feeding site itself. MBAR's Osedax project page says the worms survive by producing a root system that digests surrounding bone and releases collagen and lipids to consume. Smithsonian Ocean adds that the worms do not eat mineral bone directly. Instead, they dissolve bone material to free the fats and proteins trapped inside, and then bacteria associated with the worms help digest those nutrients. That method depends on another unusual feature, one that researchers only clarified after the original discovery. Osedax gets into bone by secreting acid. In 2013, work highlighted by UC San Diego and the Proceedings of the Royal Society, B showed that root epithelial cells express strong acid-secreting machinery, including a proton pump and carbonic anhydrase, giving the worms a way to dissolve the mineral part of the bone and expose the organic material locked inside. UC San Diego's summary of the study explains that the acid gives the worms access to collagen and lipids trapped in the skeleton, and that the root system is the tissue responsible for this bone-drilling chemistry. There is a nice unpleasant neatness to that. Bone is hard because it is mineralized. Strip away or loosen that mineral barrier, and suddenly the collagen matrix and stored lipids become reachable. Osedax does not attack the skeleton with force. It changes the chemistry around the surface it touches. The acidification step is functionally similar in broad outline to what vertebrate bone remodeling cells do in their own bodies, and the UC San Diego summary points out that similarity directly. The difference, of course, is that Osedax is not remodelling its own skeleton, it is dissolving somebody else's. That is a very specific career path, and one it appears to have followed for a very long time. Once the mineral barrier is breached, the bacteria become even more important. Smithsonian's Marine Microbes overview states that the gutless bone-eating worm Osedax frankpressi would not exist without the microbes living within its cells, because those microbes break down collagen, cholesterol and lipids from whale skeletons. More recent genomic and microbiome studies have built on that, suggesting a complex nutritional partnership rather than one simple trick. There is still discussion about exactly how nutrients move from bacteria to worm, but the central point is secure. Osedax is not working alone. Its body is built around a symbiosis, and the root system is both a feeding organ and a place where that partnership is housed. This makes the lack of a mouth less strange than it first appears. A mouth is useful when food comes in pieces that can be bitten, sucked, filtered, or swallowed. Bone on the seafloor does not arrive in a form that rewards any of those strategies. It sits there dense and locked. Osedax does not need to bite it. It needs to open it. Once you see the animal that way, the whole body plan lines up. The palps pull in oxygen. The trunk supports the visible body and reproduction. The root system penetrates the bone, alters the chemistry, and works with bacteria to release usable nutrients. The worm is less like a grazer and more like a fixed processing station set up directly on the resource. The tube around the trunk adds another useful detail. Search results from the Royal Society paper note that the trunk and palps can retract into the mucus tube secreted by the animal, and even partly into the excavated cavity in the bone. That tells you something about how exposed the worm really is. The plumes may wave outside when conditions are calm, but the animal is not simply sitting bare on the bone. It has a protective housing and a retreat. This is useful in a place where currents, sediment and visitors from the local scavenger community could all become problems. It also helps explain how something apparently delicate manages to persist on a hard, changing substrate in the deep sea. Then there is the matter of the eyes, or rather the complete lack of them. In a sunlit environment, eyes are often one of the first features people expect an animal to carry. On the deep sea floor, especially on a whale fall thousands of meters down, eyes are not necessarily worth the metabolic investment. MBAR Eyes overview says flatly that Osedax has no eyes, and that absence fits its whole way of life. A worm attached to a bone, relying on chemical and structural interactions more than visual navigation, gains little from building and maintaining visual organs. Osedax does not need to scan the horizon. It needs to stay put, breathe, reproduce, and keep opening the bone beneath it. Even so, it would be a mistake to call the animal simple. Losing familiar features is not the same as being crude. In fact, Osedax is a good example of the opposite. It belongs within the cyberglinids, a group already known for reduced digestive systems and symbioses, and it has taken that trend in a very specialised direction. The bacteria-rich root system serves the role that a gut cannot serve in this setting. The plume handles respiration efficiently. The reproductive structures are built into a fixed life on bone. A 2023 comparative genomic study cited in search results notes that the Osedax-Oceano-Spirulales partnership has unique genomic adaptations linked to bone digestion. So the worm is not stripped down because it failed to evolve certain organs. It is stripped down because different organs became more useful. That shift becomes even clearer when you look at the females in cross-section, or in developmental work. The female body is not organised around a through gut. It is organised around attachment, respiration, symbiosis, and reproduction. Search results from reproductive studies describe the oviduct emerging on the trunk and extending toward the crown of palps, while the lower body remains associated with the bone cavity and roots. Developmental and anatomical studies also show that the roots become established early after settlement, and that bacteria appear in the roots soon after. So the female does not become a normal worm and then later adopt bone feeding. The adult body is built from early on toward this exact lifestyle. The males make the contrast even sharper. In the classic form seen in many species, the large bone-boring individual is female. The males are microscopic larva-like dwarfs that live inside the female's tube and never develop the full feeding apparatus. Smithsonian Ocean notes that one study counted 111 males inside a single female, while M. Berry reports that a female may harbour several to hundreds of these tiny partners. They retain yolk reserves and focus on producing sperm, which means the female carries not just her own feeding machinery, but effectively a compact reproductive colony as well. If the female body looks unconventional at first glance, the male body looks as though development was simply told to stop early and specialise. That arrangement can sound almost absurd until you think about the environment. A rare resource appears on the seafloor, larvae arrive, space on the bone matters, food access matters. Finding a mate in open deep water is not something one wants to leave to luck. In that context, a large-feeding female with a built-in harem of tiny males is less ridiculous than it first appears. Search results from evolutionary and reproductive studies describe Osedax as the only cyboglinid genus with such pronounced sexual dimorphism, and they support the idea that larvae settling on bone become females, while later larvae settling on females may become dwarf males. In other words, even the bizarre sex difference fits the same general principle as the rest of the anatomy. Do not waste structure on anything the environment does not reward. There is one more point worth holding on to because it helps keep the animal from turning into a mere list of odd traits. Every missing feature in Osedax is paired with an alternative that solves the same problem differently. No eyes, because the adult female does not need to navigate by sight. No mouth, because food is not gathered in bite-sized form. No stomach, because the nutrient source is processed externally and with microbial help. No ordinary male-female balance, because reproduction has been reorganized around a rare, patchy habitat. The more closely scientists studied the animal, the less it looked like a creature with absent parts, and the more it looked like one with parts reassigned. And there is something almost practical in that kind of evolution. If a whale skeleton lands on the seafloor and remains there for years, then the winners are not necessarily the biggest or the fastest organisms in the neighbourhood. The winners are the ones whose bodies match the job. Osedax does. The visible plumes gather oxygen. The tube offers protection. The hidden roots open the bone and house the bacteria. The female trunk supports the reproductive system. The males reduce themselves to the minimum needed for fertilization. It is a body built not for roaming, chasing or seeing, but for settling, dissolving and staying. So when people describe Osedax as a worm with no mouth, no eyes and no stomach, they are not just offering a list of missing organs for shock value. They are describing a real reorganization of animal life around a very specific resource. The skeleton on the seafloor does not reward ordinary feeding. It rewards patience, chemistry, partnership with microbes, and a body willing to put most of its effort where the food actually is. That is why the next part of the story has to move even closer to the bone itself, because once the shape of the animal is clear, the real question is not how strange it looks, but how those hidden roots turn a hard white skeleton into dinner. Once the body plan is in place, the feeding problem becomes much easier to picture. Osedax does not roam around looking for pieces of food. It settles on a bone, secures itself there, and turns the lower part of its body into a feeding system that works directly against the skeleton. The roots are the key. MBARI describes them as a root system that digests the surrounding bone and releases collagen and lipids to consume, while primary research on Osedax roots shows that these tissues branch through cavities excavated in the bone itself. So when people say the worm eats bone, what they really mean is that it lives partly inside the bone and processes it from within. That matters because bone is not one simple material. It has a mineral component and an organic component. The mineral portion makes it hard and durable. The organic portion includes collagen and stored lipids, which are valuable food if something can get to them. The 2013 proceedings of the Royal Society bee study on Osedax roots propose that the worm first dissolves the carbonate-rich mineral matrix and then absorbs nutrients derived from the exposed bone. Smithsonian Ocean and Smithsonian's Marine Microbes pages likewise explain that the worms target fats, proteins, cholesterol and collagen locked inside whale skeletons, rather than simply scraping away at bone in the way a grazer might scrape algae from a rock. So the process begins not with chewing, but with access. If you imagine a whale vertebra lying on the seafloor after the softer tissues are mostly gone, it is still rich, but it is protected by the very thing that makes bone useful in life: stiffness and mineralization. Osedax solves that problem chemically. The roots press into the skeleton and create an acidic microenvironment. In the 2013 study, researchers found strong evidence that the root epithelium has a major acid-secreting capacity, driven by vacuolar H plus ATPase and supported by carbonic anhydrase activity. Their conclusion was that secreted acid dissolves the bone's mineral framework and allows bone-derived nutrients to be absorbed across the worm's tissues. UC San Diego's summary of the same work states the same point clearly. The worm emits acid to bore into bones and gain access to the valuable material inside. That mechanism is one of the reasons Osedax stands out even in the deep sea, where unusual feeding methods are not exactly rare. It is not just exploiting a difficult food source. It is doing so with a system that closely resembles, in broad functional terms, the way vertebrate osteoclasts dissolve bone during normal bone remodeling. The authors of the Royal Society paper explicitly point out that similarity and describe it as a case of convergent evolution. That does not mean Osedax is literally using the same tissue type as a mammal repairing a fracture. It means that when faced with the same basic material problem, both ended up relying on acid secretion as an effective way to break down mineralized bone. The roots themselves are not neat little threads poking politely into one corner of the skeleton. They can become a ramifying network. Anatomical work summarized in the Royal Society paper describes the lower trunk, ovisac, and roots lying in a cavity excavated in the bone, with the roots branching extensively. PLOS ONE's overview of shallow water Osedax similarly describes females as living anchored inside bone owing to a ramified root system extending from the ovisac. That means the visible tuft above the surface can represent a much more extensive feeding structure below, spreading through the part of the skeleton the worm has managed to open. This is also why the word roots is so useful. Even though the animal is a worm and not a plant, the term captures the direction of the feeding system. Roots go into a substrate, they branch, they create a hidden architecture that supports what is visible above. In Osedax, the roots are not drawing water and minerals from soil. They are drilling into vertebrate bone, exposing collagen and lipids, and creating a site where the worm and its microbial partners can process those resources. MBARI uses the root language directly in its project description, and the anatomical and physiological studies support it very well. Once the mineral barrier is weakened, the real nutritional reward becomes available. Bone is rich in collagen, and in large marine vertebrates it can also hold significant lipids. Smithsonian's Marine Microbes page says the microbes living within Osedax cells break down collagen, cholesterol and lipids from whale skeletons. The 2005 Symbiosis paper, which first documented the Osedax Oceanospirales association, described the bacteria as heterotrophic endosymbionts and treated the relationship as a major evolutionary innovation. Later, genomic and microbiome work has continued to support the idea that Osedax feeding is inseparable from microbial assistance, especially once the bone interior becomes chemically accessible. What is especially interesting here is that the roots are not simply chemical drills, and they are not simply sacs full of bacteria. They are both interface and processing site. The outer tissues are involved in acid secretion and likely absorption. Beneath those tissues sit specialized cells housing the symbionts. The Royal Society paper proposed that after mineral dissolution, nutrients are absorbed across the skin. Smithsonian explains that the bacteria then help digest those freed fats and proteins. More recent microbial work on bone-degrading communities has reinforced the importance of enzymes capable of working on collagen and lipids in these systems. So feeding an Osedax is best understood as a layered sequence rather than one single action. First, the bone is opened, then the internal organics are exposed, then host tissues and microbes take over the conversion of that material into usable nutrition. That sequence also explains why Osedax does not need a conventional digestive tract. A normal gut is extremely useful when food arrives in discrete pieces that can be swallowed and passed through. Osedax is working at the surface of a hard substrate that does not come apart on its own. The food is not something that needs to be taken inside first. It needs to be unlocked where it sits. MBAR his Osedax page makes this very point in simpler form, stating that the worms lack mouths and guts, but survive by producing a root system that digests surrounding bone and releases collagen and lipids to consume. Once you accept that arrangement, the absence of a mouth or stomach stops looking like a missing feature and starts looking like an unnecessary expense the lineage was able to drop. There is still some uncertainty in the fine details, and it is worth keeping that uncertainty rather than smoothing it away. Smithsonian Ocean notes that exactly how the worm acquires nutrients from the bacteria is not fully known. One possibility is that the worm digests some of the bacteria, another is that nutrients are transferred from the bacteria to the worm in some other way. The broader picture is secure, but the precise bookkeeping at the microscopic level is not completely settled. That is often the case in symbiosis research. Scientists can clearly see that a partnership is essential and can identify major metabolic roles on each side, but the exact exchange rates, timing and transport steps can take much longer to pin down. What we can say with confidence is that the feeding system is extremely specialized. The acid-secreting machinery is strongly expressed in the roots. The symbionts are specifically associated with those feeding tissues. The worm is fixed to a substrate that most animals cannot exploit after the easy flesh is gone, and the outcome is substantial enough that Osedax can strongly influence how long bones persist at whale falls. M.B. Yari's whale fall work has argued that these worms largely control the fate of whale falls in Monterey Canyon because they consume the bones, which are the primary remaining source of nutrition at that stage. In other words, their feeding method is not a minor sideshow in the life of a whale fall. It is one of the processes that decides how the later stages unfold. There is a practical side to the chemistry as well. Acid secretion is not free. It requires metabolic energy, oxygen, and supporting cellular machinery. That helps explain why the exposed palps matter so much. The feeding roots may be doing the dissolving and absorbing, but they depend on the rest of the worm to keep oxygen and circulation going. Studies on Osedax physiology and anatomy have emphasized the vascularized nature of the root system and the respiratory role of the palps. This makes the worm feel less like a simple driller and more like a carefully integrated machine. The plume handles gas exchange, the body distributes resources, the roots handle access and uptake. None of those pieces makes full sense in isolation. Another detail that helps is scale. Osedax does not need to reduce an entire whale skeleton in a hurry. It works locally and persistently. A single female opens and inhabits a small cavity. Many females together can cover a bone surface, each producing its own borehole and root network. Fossil studies and modern observations both show that the traces left by Osedax can be dense and characteristic, because the worms do not merely sit on the outside. They leave behind evidence of penetration into the bone. Search results from paleontological work and newer fossil trace discussions both tie those borings directly to the acid-based root system, first documented physiologically in living worms. That slow, local feeding style also fits the resource itself. A whale skeleton is a big structure, but it is not all equally available at once, and it may persist for years depending on conditions. Osedax does not need a fast strike. It needs persistence. It needs the ability to stay put, keep dissolving, keep branching, and keep drawing nutrition from the cavity it has made. PLOS ONE's review of shallow water Osedax and Barry's research summaries both emphasise the anchored female lifestyle and the root system's role in maintaining access to the bone. This is feeding as tenancy. The worm does not visit the food, it moves into it. There is also a good reason Osedax ended up as a siboglinid solution rather than a standard predatory one. Siboglinids are already known for relying on symbiosis and for reducing or losing the ordinary gut. In that context, Osedax looks less like a shocking exception and more like a very specialised extension of a family tendency. The 2005 paper on evolutionary innovation framed Osedax as a new heterotrophic symbiosis within a group better known for other nutritional arrangements, while later genomic work suggests the symbionts complement host metabolic deficiencies in ways closely tied to bone-based feeding. The root system is therefore not just a novel organ by itself, it is part of a bigger shift in how the entire animal economy is organised. When you put all of that together, the feeding process becomes surprisingly clear. A larva settles on bone and develops into a female. The female produces a tube, trunk, palps, and the beginnings of the hidden lower system. The roots spread into the skeleton. Acid secretion from the root epithelium dissolves the mineral component. This exposes the collagen and lipids. Those nutrients are then absorbed and processed with the help of intracellular bacteria associated with the roots. The worm remains fixed in place and continues this work over time, while many neighbouring females may be doing the same across the same bone. Each step has been supported by either anatomical, physiological, or symbiosis-focused work, even if the exact nutrient transfer details are still being refined. And perhaps the most useful thing about understanding the feeding is that it removes the temptation to think of Osedax as merely grotesque. Bone-eating worm is a phrase that invites a certain kind of reaction, but the reality is less theatrical and more exact. This is an animal specialised for a resource that most others cannot use at that stage of decomposition. It does not crunch bones, it does not tear them apart by force, it dissolves, infiltrates, absorbs, and partners with microbes. It turns a hard, nutrient-rich structure into a workable food source through patience and chemistry. Smithsonian's resources and MBARE summaries consistently present that as the core of the worm's biology, and the 2013 acid secretion study gave the mechanism a firm physiological basis. There is even something a little humbling in how indirect the whole meal is. From our everyday point of view, eating often means taking in something obvious, chewing it, swallowing it, and being done. Osedax reminds us that nutrition can be a distributed process spread across tissues, symbionts, and a modified environment. The meal is not just inside the worm. The meal is partly in the cavity, partly in the root epithelium, partly in the bacteria, and partly in the chemistry happening at the surface of the bone. That is exactly why the next part of the story belongs not just to the worm, but to the bacterial partners tucked within those tissues, because once the roots have opened the skeleton, the feeding system is no longer only the worm's work. If the roots are where the feeding happens, then the bacteria are the reason that feeding works as well as it does. Osedax is not simply a worm that drilled its way into a useful resource and figured everything else out alone. It is a host. Beneath the skin of its root tissues are specialised cells called bacteriocytes, and inside those cells live bacterial partners that help process the organic riches sealed inside vertebrate bone. Nature Communications describes Osedax as having bacteriocytes concentrated in the subepidermal connective tissue of the lower trunk that grows directly into bone, while the 2015 PLOS ONE review says these Oceanospirillales gamma proteobacteria are housed in bacteriocytes within the roots and degrade organic compounds sequestered in the bone. Smithsonian's Marine Microbes overview puts it more directly still. Osedax francpresi would not exist without microbes living within its cells that break down collagen, cholesterol and lipids from whale skeletons. That arrangement matters because Osedax has given up the usual digestive route. It has no mouth and no gut. So there is no long internal tube taking in food in pieces and reducing it step by step. Instead, the worm solves the problem by building a feeding organ that is also a living home for microbes. The roots open the door, and the bacteria help handle what lies behind it. Berry explains that the root system digests surrounding bone and releases collagen and lipids to consume, and that those same roots house symbiotic bacteria that appear to play a crucial role in nutrition. Nature Communications adds another important detail. The roots express high levels of proton pumping and carbonic anhydrase machinery, indicating that acid is used to dissolve the bone matrix so collagen and lipids can then be absorbed across the root epithelium. In practical terms, the worm does not just sit on the bone with bacteria tucked away for company. Host and symbionts are built into one working system. It helps to picture where those bacteria actually are. They are not just floating around loosely inside the female. They are packed in specific tissues, close to the place where the worm is meeting the bone. Search results from the early symbiosis work describe Oceanospirillales type bacteria densely populating the ovisac and root tissues and appearing concentrated in bacteria sites, and the 2013 acid secretion paper identifies the roots as the extensions that penetrate bone and host heterotrophic symbiotic bacteria beneath the epidermis. That phrase, beneath the epidermis, is useful because it gives the right sense of depth. The bacteria are not surface grime, and they are not casual hitchhikers clinging to the outside of a tube. They are integrated into the body, living just under the worm's own outer layer, in the tissues most closely tied to feeding. That is why this part of the story belongs under the skin rather than beside the worm. The first researchers to describe this relationship recognized right away that it was not a standard cyberglinid arrangement. In 2005, work on Osedax framed the association as a new heterotrophic marine symbiosis with Oceanospirillales, which was important because many other famous cyberglinids rely on chemosynthetic bacteria for energy. Osedax was doing something different. Its bacteria were not using sulfur or methane to build a food web the way vent and seep symbionts do. They were associated with a host feeding on bone-derived organic material. The 2023 comparative genomic study makes this distinction very clear, showing that the Osedax Oceanospirillales partnership follows a different genomic route from the symbioses seen in vestimentiferen tube worms. In other words, even within a family already known for unusual microbial partnerships, Osedax had taken its own route and built a different kind of alliance. That difference shows up in what the bacterial partners are thought to do. Smithsonian says the microbes break down collagen, cholesterol and lipids from whale skeletons. The +1 review adds that these chemoorganoheterotrophic bacteria degrade the organic compounds retained in bone once the inorganic matrix has been dissolved. Nature Communications pushes the picture further by linking the symbiosis to bone digestion at the genomic level. The Osedax host shows losses in pathways for amino acids that are abundant in vertebrate bone. The endosymbiont has the glyoxylate cycle that could help turn bone-derived lipids into carbohydrates, and the host has expanded matrix metalloproteases that could aid bone digestion. This matters because it suggests the partnership is not a simple case of worm opens bone, bacteria finish dinner. The host and symbiont may each be handling different parts of the nutritional job, with the whole system tuned to a food source rich in collagen and lipids. That does not mean every detail is settled. In fact, one of the more interesting things about Osedax is how clearly researchers can see the importance of the microbes while still working out the fine print of the exchange. Smithsonian's Zombie Worms Crave Bone page notes that after acid frees the trapped fats and proteins, symbiotic bacteria living in the worm's body digest those nutrients. But it also says scientists do not yet fully understand exactly how nutrients move from bacteria to worm. A recent fossil trace paper, discussing the modern biology for context, makes the same point in another way by noting that the precise mode of nutrient transfer from symbiont to host, whether through direct provisioning or digestion of symbionts, still needs fuller study. That uncertainty is not a weakness in the story. It is just a reminder that some of the most important partnerships in biology are obvious in outline long before every microscopic transaction has been counted. There is another useful question here, and it is one researchers have taken seriously. Where do the bacteria come from in the first place? Are they handed down directly from parent to offspring, or picked up from the environment? The evidence points strongly toward the second option. The 2015 PLOS ONE review states that these Oceanospirillale symbionts are horizontally transmitted. A 2012 study, summarized in search results, says Osedax acquires its essential endosymbionts locally from the environment in which the larvae settle, and a 2010 symbiont diversity study reported patterns consistent with repeated infection events as host root tissues grow into whalebone. That makes ecological sense. A larva drifting through the sea cannot carry a ready-made bone microbiome that knows every local condition it will meet. Picking up suitable partners from the surrounding environment gives Osedax flexibility, though it also means the partnership must be rebuilt each generation, and that flexibility seems to come with diversity. Researchers have not found one perfectly uniform bacterial companion repeated without variation in every worm. The 2010 study on Osedax muciflorus reported a higher diversity of endosymbionts than previously recognized, with sequences falling into multiple rebo species, while the 2012 Monterey Bay time series study followed symbiont diversity across 9 Osedax species over 3 years at an experimental whale fall. That same 2012 work emphasized temporal variation and a lack of strict host specificity among the bacterial endosymbionts. This is worth slowing down for because it makes the symbiosis feel less like a locked marriage between one worm species and one bacterial species, and more like a structured but somewhat flexible partnership assembled from a pool of related Oceanospirillales available at the site. The worm still needs the right kind of helper, but apparently not always the exact same bacterial identity down to the smallest possible label. That in turn changes how we should imagine the root tissues. They are not just fixed organs growing larger in a passive way. They are growing surfaces that may be colonized and recolonized as they expand into the bone. The 2010 symbiont diversity work specifically interpreted its results as consistent with repeated infection events as roots grow into whalebone. The 2012 study likewise focused on temporal dynamics, showing that the microbial makeup associated with Osedax can vary over time at a whalefall site. So the worm's hidden feeding system is not static. It is a place where tissue growth, bone penetration, chemistry, and microbial acquisition continue together. If the upper part of Osedax looks calm, the lower part is busy in a way we do not get to see directly. Beneath the visible plume, the animal is extending its reach and stocking the right workers at the same time. There is also a newer twist that makes the bacterial story even less tidy in the best possible way. Osedax is known mainly for its intracellular Oceanospirillales endosymbionts in the roots, but recent work has shown that there can also be a dynamic community of external bacterial associates on the worm. A 2023 MBO study reported a persistent succession of Campylobacteriales epibionts integrated into the epidermis of Osedax over a 14-year period, shifting as whale decomposition progressed on the sea floor. That does not replace the root symbionts as the main feeding partners, it broadens the picture. The worm is not simply carrying one bacterial population in one hidden tissue and nothing else. It appears to participate in a changing bacterial neighbourhood, with internal partners tied to nutrition and additional surface associates responding to the carcass as conditions change around it. That broader microbial setting makes a lot of sense when you remember what a whalefall is. It is a changing chemical landscape. Early on there is plenty of soft tissue and plenty of scavenger activity. Later, the bones dominate. Later still, sulfide and other reduced compounds can shape what sorts of microbes and invertebrates thrive nearby. Osedax lives through part of that long transition, and its microbial companions seem to reflect it. The 2023 Epibiont study ties the Campylobacteriales succession directly to different stages of whale decomposition, while earlier Whaleful work and MBARI's own summaries emphasise that bones become available only after scavengers strip the carcass, and that Osedax then moves in on the remaining resource. So the bacteria beneath the skin are part of a much larger story, in which the chemistry of a dead whale keeps changing for years, and the worm is both a resident of that process and a force helping to push it along. The genomic work from 2023 also makes the relationship look more intimate than a simple convenience arrangement. Nature Communications reports that O. frank presi has a reduced gene repertoire compared with vent-dwelling vestimentiferae, and that the endosymbiont appears able to complement host metabolic deficiencies, including pathways related to nutrient use in a bone-rich environment. That does not mean the worm is helpless without any one particular bacterium from birth, because the evidence still supports horizontal acquisition. It means that once the partnership is established, the host seems to lean into it at the genetic level. Bone is such a specialised food source that the host and symbiont together appear more efficient than either would be alone. This is the kind of arrangement evolution tends to refine over a long time. One side losing what the other side can supply, both sides narrowing toward a shared niche, until the whole system begins to look less like cooperation and more like a merged way of life. It is tempting at this point to turn the bacteria into invisible assistants and leave them there, but that undersells them. They are not background support. They are part of the feeding organ. They are part of the nutrient pathway. They are part of the reason Osedax can exploit bone at all. Smithsonian says the worm would not exist without them. MBARI says they appear to play a crucial nutritional role. The PLOS One review states plainly that Osedax nutrition is obtained through this symbiosis. When several different lines of evidence keep returning to the same conclusion, it is usually a good idea to believe them. The worm is strange on its own. The worm plus bacteria is the actual animal doing the work on the seafloor. So if you were to look at an Osedax female rising from a whalebone, the feathery crown is only the visible half of the story. Down below, under the outer tissues, the roots are spread through the cavity they have made. Acids are loosening the mineral matrix, organic material is being exposed, and bacterial cells in bacteriocytes are handling compounds the worm cannot efficiently manage alone. The whole arrangement is hidden, local, and patient. Nothing about it is dramatic in the way a predator strike is dramatic, but it is extremely effective. And once you see that, the whalebone stops looking like a hard object with a worm attached to it and starts looking like a living work site, which is a useful way to hold the image in mind before we pull back again and look at the larger setting that makes a partnership like this worth evolving in the first place. Those bacteria beneath the skin only make sense because the food source is large enough, rich enough, and persistent enough to reward that kind of extreme specialization. A whale fall is exactly that. In the deep sea, most animals live on a very slow rain of organic matter sinking down from the surface, often called marine snow. It keeps the system running, but usually in small installments. A whale carcass changes the scale completely. NOAA explains that when whales die and sink, they create a sudden, concentrated food source, and a recent review describes whale falls as deep-sea oases in an environment otherwise poor in energy and nutrients. That same review notes that the soft tissue of a 30-ton whale can contain about 1.2 times 10 to the power of 3 kilograms of active organic carbon, roughly equivalent to the background organic carbon flux to 100 square meters of deep sea floor over 1,000 years. That is why the word banquet is not exaggerated here. This is not a slightly better meal than usual. It is a major delivery of food to a place where food normally arrives slowly and thinly. The Frontiers review says whale carcasses may transport organic matter to the deep sea about 2,000 times faster than the normal supply from marine snow, and it describes whale falls as important to biodiversity, succession, and the ocean carbon cycle. So when a whale reaches the seabed, it does not just feed a few lucky scavengers, it changes the local economy of life for a long time. It also helps that whales are built in a way that makes this possible. They are large, full of energy-rich tissue, and their bones themselves remain valuable after the flesh has been stripped away. NOAA notes that the skeleton can support rich communities for years to decades, both as hard substrate for colonizing invertebrates and as a source of sulfides generated by the decay of organic compounds in the bones. The Frontiers Review goes even further and says a whalefall can support a decomposer-dominated ecosystem for up to 100 years, depending on conditions. So this is not a brief flash of abundance followed by immediate silence. It is a long-staged event. The first stage is the most obvious and, in a way, the least mysterious. Large mobile scavengers arrive and remove the soft tissue. The Monterey Canyon whalefall study says that this mobile scavenger stage is usually made up of about 40 species, mostly fish and crustaceans, and that they can remove more than 90% of the soft tissue, usually within about 4 months after the carcass reaches the sea floor, though timing depends on carcass size and depth. NOAA gives the same broad picture in simpler terms, noting that scavengers consume the soft tissue in a matter of months. This is the part people usually imagine first: sharks, hagfish, crabs, and other consumers making short work of the easy food. Even here, though, it is worth slowing down and not turning the scene into a dramatic feeding frenzy from a nature documentary trailer. A whale fall in the deep sea is busy, but it is not busy in the same way a kill on land is busy. There is no daylight, no circling vultures, no dust, no noise in the ordinary sense. The carcass lies on the bottom, and animals come and go through cold water and darkness. The scale is huge, the pace can be uneven, and much of the action is chemical as much as biological. Tissue is eaten, yes, but organic fragments also spread into the surrounding sediment, and microbes respond almost immediately to the new material. The banquet is never just about mouths. It's also about what leaks, settles, enriches, and begins to alter the seafloor around the carcass. NOAA says the detritus enriches nearby sediments for over a year, and the Monterey study describes the development of large microbial mats and local organic enrichment around the bones. That leads into the next stage, often called the enrichment opportunist stage. The Monterey Canyon paper says it may last as long as 2 years, and is typically made up of around 20 species, mostly polychaetes and crustaceans. These are animals that thrive in the enriched sediments and leftover organic material associated with the carcass. The most important point is that they are not necessarily feeding on fresh flesh in the obvious way the first scavengers were. They are taking advantage of what the whale has done to the patch of seafloor around it. The carcass has turned an ordinary area of deep sediment into a richer, messier, more active place, and that opens the door for a different cast of residents. This is one of the reasons whalefalls are so valuable to ecologists. They make succession very easy to see. Instead of one resource supporting one set of consumers and then disappearing, the carcass supports a sequence of communities, each one tied to a different stage of breakdown. Noah summarizes this by saying that different stages in whale decomposition support a succession of marine biological communities, and the Frontiers Review makes the same point, calling whalefalls important for understanding ecosystem succession in deep-sea desert areas. So the whalefall is not one banquet with one seating. It is a long reservation schedule, and the guest list changes as the meal changes. Then comes the stage most closely tied to Osedax and to the strange bacterial partnerships we have just been talking about. In the Monterey framework, this is the sulfophilic stage. The paper explains that it relies on the coupling between decomposition of bone lipids and seawater sulfate reduction, and that it can be dominated by microbial mats plus more than 100 invertebrate species, including sulfide-tolerant and sulfide-dependent mollusks. It may last for years and even decades, depending on the size of the whale. Noah echoes the same broad pattern, noting that whale bones become a source of sulfides from the decay of organic compounds, and that microbes using energy from these reactions can support entire deep-sea food webs for as long as the food source lasts. This stage is one of the most quietly impressive parts of the whole process. To most of us, bone looks like the final hard remnant after the important nutrition is gone. In a whale fall, that is simply not true. The skeleton is still rich, especially because large whales store lipids in their bones. The Frontiers Review notes that whale falls create sulfur-rich habitats not only because of the lipid-rich skeleton itself, but also because of the surrounding sediment chemistry and that sulfate reduction and sulfide levels around a 30-ton whale carcass can remain elevated for at least 7 years. So the skeleton is not dead weight. It is a long-term energy reserve, and that reserve feeds not only Osedax but also microbial and invertebrate communities that depend on sulfide-rich conditions. That is where the phrase sudden banquets becomes especially useful. The whale arrives suddenly, but the feast unfolds over time. There is an immediate pulse of abundance for large scavengers, then a longer enrichment of the surrounding sediments, then a still longer period in which the skeleton itself supports specialized life. It is sudden at the beginning and prolonged afterward. It is a shock to the system followed by a long period of ecological bookkeeping. And because the deep sea is normally so sparse that bookkeeping matters, the Frontiers Review says whale fall species richness can reach 407 species, slightly lower than hydrothermal vents, and much higher than cold seeps in the comparison cited there. Whether or not one wants to focus on the exact numbers, the larger point is clear. A whale fall can support a remarkable amount of life for a very long time. It also matters where these carcasses occur. A single whale fall can be thought of as an oasis, but over long time scales whale falls may act as a scattered network of habitat islands across deep basins. The Frontiers Review says whale falls may contribute to the dispersal of thiophilic species between sulfur-rich habitats such as hydrothermal vents, cold seeps, and anoxic basins. Earlier whale fall work, as summarised in the Monterey paper, had already raised the idea that such sites could play a significant role in regulating abundance and diversity in deep regions. So a whale fall is not just an isolated feast. It may also be one stop in a much larger pattern of dispersal and specialisation. For larvae drifting through the deep sea, a carcass is not merely food. It is a chance to settle, grow, reproduce, and perhaps send the next generation onward when the time comes. This is one reason the decline of large whales during industrial whaling mattered far below the surface, even though the dead animals themselves were never seen by most of the people doing the hunting. The Frontiers Review states that whale falls are important geographical stations for the evolution and development of life in extreme deep-sea environments, and its corrigendum notes estimates that human whaling may have reduced whale fall habitats by as much as 95%. Potentially affecting biodiversity across connected deep-sea systems. That idea should probably make anyone pause for a moment. It means the removal of whales from the oceans was not only a loss in the living surface world, it also likely reduced the number of these deep-sea banquets, and therefore the habitat available to the specialised organisms that depended on them. At the same time, modern discoveries are still reminding researchers how much remains hidden. NOAA Fisheries reported in January 2025 that sonar surveys in the San Pedro Basin off Southern California documented at least 6 whale falls directly and suggested there may be more than 60 in an approximately 350 square mile area, potentially one of the greatest concentrations of whale falls ever found if confirmed. NOAA also noted that these remains may help scientists explore the role of whale falls in carbon capture and climate mitigation. So even now, after decades of whale fall research, the seafloor is still capable of producing the sort of result that makes people sit up and recalculate how common these sites may be in some places. That newer finding also helps correct a common mental picture. It is easy to imagine whale falls as impossibly rare, one skeleton in a vast dark emptiness, almost mythical in their scarcity. In truth, they are certainly patchy and hard to find, but not necessarily singular or irrelevant. If a basin can hold dozens of whale falls, then for the animals adapted to exploit them, the deep sea may be less like a featureless desert, and more like a landscape with occasional, unevenly spaced but very meaningful stations of abundance. Researchers still need to work out how many such concentrations exist, and how long the remains in San Pedro Basin have been there, but the basic point stands. The banquet sites may be more common in some settings than human observers once guessed. For Osedax, all of this is excellent news in the strictly biological sense. A whale fall arrives with more food than most deep-sea organisms could hope for in one place, and that food remains useful in stages. Osedax does not need the first rush of soft tissue. In fact, it does better once that stage is mostly over, and the bones stand exposed as the main remaining reserve. The Monterey study explicitly places Osedax in the later part of whalefall succession, associated with the enrichment opportunist and sulfur-philic stages, and Noe notes that skeletons can support rich communities for years to decades. That gives a bone-eating specialist time to settle, grow, reproduce, and turn the skeleton itself into food and habitat. The banquet, from Osedax's point of view, begins late and lasts a long time. There is also something useful in remembering just how large the original event is. The deep sea usually runs on fragments. A whale fall delivers whole architecture. Soft tissue feeds one guild of consumers. Detached organic matter enriches sediment for another. Bone lipids and bone proteins support later microbial and invertebrate communities. The skeleton becomes both pantry and platform. That layered use of one body is why whale falls have become such a powerful example in marine ecology. They show, in a particularly visible form, how one massive pulse of organic matter can be redistributed across time, across taxa, and across feeding strategies. Noe and the Frontiers Review both emphasize that the carcass adds to the ocean food chain by creating a new food web in deep water. It is a neat reversal of what many people assume about the deep ocean. From the surface, the abyss can seem empty, almost static, as though very little happens there except slow settling and slower decay. Whale falls reveal the opposite. The deep sea is ready, the scavengers are ready, the opportunists are ready, the sulfide-based communities are ready, Osedax is ready. The banquet does not create life from nowhere. It reveals how many forms of life are already prepared to respond the moment a suitable resource appears. The suddenness belongs to the delivery, not to the adaptations. Those were waiting long before the whale arrived, and perhaps that is the most important thing to carry forward from this part of the story. A whale fall is not just a dead animal resting on the seabed. It is a long ecological sequence in which abundance arrives abruptly and is then divided, recycled, and specialized over years or decades. It begins with large scavengers and ends with bone, microbes, sulfides, and organisms able to use what almost nothing else can. Once you see that, Osedax stops looking like an isolated oddity attached to a random skeleton. It starts to look like exactly the sort of animal a place like this would eventually produce, which is where the story naturally turns next, because among the many strange residents of a whale fall, few have family arrangements as peculiar as the bone-eating worms themselves. If Osedax was strange only because it eats bone, that would already be enough to keep most people interested for a while. But then you look at its family arrangement, and things take a sharp turn into territory that sounds less like ordinary marine biology and more like someone quietly trying to win a contest for the most peculiar domestic setup in the ocean. The large individual fixed into the bone is usually a female. She grows the plume, the trunk, the ovisac, and the branching roots that carry out the real work of feeding. The males in most known species do not become anything like that at all. They remain microscopic, live inside the gelatinous tube around the female, and never feed on bone. MBARI says that, with one exception, known Osedax species form harems of dwarf males inside the female's tube, and that some large females have been found carrying more than 600 of them. PLOS ONE likewise describes the usual pattern as marked sexual dimorphism, with paidomorphic dwarf males hosted within the lumen of the female's tube. It is difficult to overstate how odd that is by ordinary standards. We are not talking about males that are simply smaller or a little more delicate or less dramatic in appearance. We're talking about males so reduced that for a while, after the first discovery, scientists were not even seeing obvious adult males at all. The original science report on Osedax described skewed sex ratios with numerous dwarf males living in the tubes of females, and later developmental work has reinforced the point that these males are microscopic compared with the females that dominate the bone surface. The 2024 Frontier study says Osedax is the only genus in Symbiotic that shows this kind of sexual dimorphism, and that in the usual pattern, females have the full adult body plan while males remain microscopic and non-feeding. What makes that even more striking is that the female is not small. Depending on species, she can extend several centimetres in length, with a trunk rising from the bone and roots buried beneath it, while the male looks more like a larva that stopped developing early and decided that producing sperm was a perfectly respectable endpoint for a life. That is not just a colourful way to put it. The 2015 Current Biology paper on Osedax praepus states directly that in the typical pattern, Osedax males are developmentally arrested larvae that produce sperm from yolk reserves. The 2024 developmental study then goes further and argues that male dwarfism in Osedax is a direct result of progenesis, meaning sexual maturation happens while the rest of the body remains larval and somatic development stops early. In plain language, the male stays tiny because it reaches functional adulthood before building the larger adult features seen in females. That alone would be enough to make Osedax memorable, but there is more. These dwarf males do not live out on the bone as independent little residents. They live inside the female's tube. The tube then is not just housing for the female trunk, it becomes a packed reproductive chamber, holding the female herself, her resident males, and in some species and observations, embryos and larvae at different developmental stages as well. The 2015 PLOS ONE study of shallow water Osedax reported embryos and larvae at different stages inside the lumen of female tubes, with a single tube containing as many as 81 embryos or larvae. That means the female's tube can serve all at once as home, mating site and nursery. There are family homes, and then there is whatever this is. One of the most useful ways to understand this arrangement is to stop measuring it against human ideas of fairness or symmetry, and start measuring it against the deep sea. A whale bone is a rare and limited patch of good habitat. Space on the bone matters. Food access matters. Finding a mate in a dark, vast environment where individuals may be sparse is not something any lineage wants to leave to chance. MBRI explicitly says male dwarfism in Osedax is thought to be advantageous, where finding mates is difficult, as in sparsely distributed deep-sea species, and where males and females would otherwise compete for the same limited resource, namely the bone itself. The 2024 Frontiers paper makes the same ecological point in broader terms, noting that dwarf males can reduce competition between sexes in habitats with limited food or space, and can improve fertilization when mating groups are small or dispersed. Once you look at it that way, the logic becomes almost irritatingly sensible. The female needs space and access to bone. She is the one doing the expensive work of growing the full body, maintaining the root system, housing the symbionts, and producing eggs. The males do not need roots, or plumes, or a large trunk, because their job is not to feed. Their job is to stay close to a female and make sperm. A tiny male living in the tube solves several problems at once. He does not compete much for space, he does not compete for the bone, he does not need to go searching for a mate across the seabed. He is already in the right place. A family arrangement that sounds absurd from a distance turns out to be exactly the sort of thing evolution produces when the habitat is scarce, and the dating scene is, to put it mildly, not lively. Scientists work this out piece by piece. Early on, after Osedax was first described, a common hypothesis was that sex might be environmentally determined. The idea was straightforward enough. Larvae settling directly on bone would develop into females, while larvae landing on a female would become dwarf males. That view appears in the original 2004 science paper, and in later discussions of Osedax biology, and it fit the broad pattern people were seeing on the bones. The 2008 molecular ecology study then added an important twist by showing that the males in female tubes were recruited from a common larval pool rather than representing only close kin from a single mother, which argued against a highly inbred household and suggested continuing recruitment from the surrounding plankton. But that is not the end of the story, and this is where more recent work becomes especially important. In 2025, an Evo study on Osedax japonicus reported evidence that larvae are already sexually determined before metamorphosis, rather than being assigned sex entirely by environmental cues after settlement. The researchers found that half of the late larvae carried a transient gut, identified that gut-bearing larvae were females through sex-specific gene expression, and concluded that sexually biased gene expression and a roughly 1-to-1 sex ratio indicate pre-metamorphic sexual differentiation. In their discussion, they say this evidence refutes the previously accepted hypothesis of environmental sex determination, at least for O. japonicus. That means the scientific picture has shifted. The old view was elegant and plausible, but newer developmental evidence suggests the story is more complicated and more predetermined than people first thought. That change is worth including, because it makes the family story more interesting rather than less. It tells us that the males do not merely become males because they happen to land in the wrong place. At least in O. japonicus, they appear to be male already, even before the body begins to show obvious adult dimorphism. The 2025 study found that female larvae carry a transient gut, and males do not, and that sex-biased gene expression appears as early as the 2-day larval stage. So what settlement changes may not be sex itself, but the root development can take from there. A female larva settling on bone can proceed into the full, bone-boring adult form. A male larva does not. That makes the deep-sea family arrangement look less like a simple environmental switch, and more like a carefully timed developmental fork built into the life cycle from very early on. The males themselves remain extraordinary. They're often described as pedomorphic, meaning they retain larval features into the reproductive adult state. The 2024 Frontiers paper notes their superficial resemblance to larvae in size, anterior ciliation, and the number of chaetae, and argues that their dwarfism results from progenesis, an early cessation of somatic growth paired with sexual maturation. The 2015 Current Biology paper says much the same in shorter form, calling them developmentally arrested larvae that produce sperm from yolk reserves. It is a very efficient piece of engineering if all you need is a male that can fertilize eggs in a small tube. It is also a rather humbling reminder that an adult body is not a fixed package. Evolution can stop development halfway, keep the reproductive parts, and call the result good enough. And it really is good enough, because these tiny males appear to do the job in large numbers. Mbare says females accumulate more males as they grow, and some harems exceed 600 males. The 2004 science report and later anatomical work described numerous dwarf males in the female tube, while reproductive studies have found sperm within female reproductive tissues, supporting the idea that fertilization takes place in intimate proximity, rather than as some long-distance gamble in open seawater. The family arrangement is strange, but not haphazard. It is compact, crowded, and geared toward making sure that a sessile female attached to a rare bone does not have to wonder whether a suitable partner will ever arrive. There is also something quietly funny about the balance of investment here. The female gets the plume, the trunk, the roots, the symbionts, the full feeding machinery, and the main role in turning bone into usable nutrition. The male gets what looks, from a harsh but fair point of view, like the minimum viable body plan for sperm delivery. It is hard not to admire the economy of it. If there were a deep-sea award ceremony for refusing to spend extra energy on unnecessary features, Osedax males would at least make the shortlist. though one suspects they would not stay long enough at the reception to enjoy it. Then again, evolution is rarely content to leave even a bizarre arrangement alone forever. There is a major exception in the genus, and it complicates the story in exactly the way good biology often does. In 2015, researchers described Osedax priapus, a species in which males are no longer microscopic tube dwellers. Instead, both sexes are bone-eating adults of more similar size, and the males possess roots, symbiotic bacteria, and a trunk that allows them to move across the bone and inseminate females. The paper calls this a dramatic evolutionary reversal in sexual size dimorphism, and MBARI describes it as a throwback to a more ancestral condition, with large bone-eating males replacing the usual dwarf system. So even within Osedax, the strangest family in the ocean still has one relative who looked at the household rules and went in a very different direction. That exception actually helps the usual pattern stand out more clearly. It shows that dwarf males are not an unavoidable mechanical consequence of being an Osedax. They are an evolved strategy, one that can in principle be reversed. The 2015 paper places Opriapus well within the Osedax clade and treats its larger males as a secondary shift away from the typical condition. The 2024 Frontier study says the species exemplifies a secondary gain of macroscopic male size and sexual monomorphism. In other words, the common Osedax arrangement is not the only possible way to be a bone-eating worm family. It is just the one most species settled on, presumably because it works very well under the usual deep-sea conditions of patchy habitat and limited mating opportunities. Still, for most species, the standard pattern remains almost comically one-sided. A female anchored in bone may be several centimetres long. The males in her tube are microscopic. The same female may hold embryos and larvae as well. The PLOS-1 study reports larvae lacking bacterial symbionts and packed with yolk, while Mbari emphasises that females continue to accumulate dwarf males as they grow. So the tube is not a static arrangement assembled once and left alone. It is a dynamic little household with recruitment, reproduction, and development all pressed together in one narrow space around the female body. That may be one reason Osedax feels so memorable to people. It takes a life cycle that is already odd and then folds it into architecture that is even odder. There is no need to force this into human family language too far, because the biology is already doing enough on its own. But it is hard not to notice the broad pattern. The female is the household, the food supply, and the long-term resident. The males are tiny, numerous, and resident in her tube. The next generation begins nearby. The bone supplies the base. The whole arrangement is built around a resource that is rich but finite, rare but worth finding, and impossible to exploit casually. If a lineage were ever going to produce a reproductive system that looks eccentric to us and perfectly practical to itself, this was the place. And the really useful thing about understanding this family arrangement is that it sets up the next question almost automatically. Once you know that a female can sit on a bone with a crowd of microscopic males in her tube, and once you know that larvae are produced into a world where the right kind of bone patch may be many meters, kilometers, or even more away, then the obvious thing to wonder is how the next generation ever finds its way there at all. Because a family this strange only continues if the larvae can leave one hard meal in the dark and somehow locate another, which is where the story naturally shifts next, out of the crowded tube and back into the water column, where tiny drifting bodies begin the long search for bone. Once the next generation leaves that crowded female tube, the whole story changes in scale. Inside the tube, Osedax life is cramped, local and organised. Out in the water, it becomes a problem of distance, timing and luck. A larva is tiny. The seafloor is huge. Suitable bones are patchy. The adults cannot swim off and inspect the neighbourhood. So the job of finding the next meal and the next place to live falls to a drifting stage that has to manage the search before its own limited resources run out. Studies on Osedax development describe the larval stage as lessitotrophic, meaning it depends on yolk rather than feeding. and recent work on Osedax likewise describes the larvae as non-feeding with rapid early development. That detail matters immediately, because a non-feeding larva does not get to improve its odds by stopping for snacks. It goes out into the water with what it already has. The yolk packed into the egg is the travel budget, the emergency reserve, and the final warning that the trip cannot go on forever. PLOS 1 notes that once fertilised, oocytes are released and give rise to lithotrophic trochophore larvae that allow dispersal and colonisation of new substrates, while the 2013 developmental study on Osedax reported that larvae survived for 10 days under laboratory conditions. 10 days is useful, but it is not generous when the habitat consists of scattered bones on a dark seafloor. The deep sea does not exactly place glowing arrows over vacant whale vertebrae. Early development appears to move quickly. PLOS 1 cites earlier work showing that fertilised oocytes released through the oviduct developed into early trochophores after 2 days under laboratory conditions, and the 2024 developmental study describes embryos becoming non-feeding trochophore larvae within a few days at 10 degrees Celsius. So Osedax does not linger for long in some prolonged, leisurely juvenile ambiguity. The larvae form, enter the water column, and begin the short period in which drifting and settlement have to succeed. That is one reason the whole dispersal stage feels a bit tense, even if the animal itself gives no outward sign of panic. The clock is running from the beginning. What the larva does not carry is almost as important as what it does. Osedax eggs and pre-settling larvae are aposymbiotic, meaning they do not already contain the essential bacterial partners that the adult female uses in her root tissues. The 2012 BMC study says this directly and adds that the worms acquire those endosymbionts locally from the environment in which their larvae settle. That means the larva is not simply looking for a place to become an adult, it is looking for a place where the full adult feeding system can be assembled afterward. First the bone has to be found, then settlement happens, then the root system develops, then the bacteria are acquired. It is a staged transition, not a case of a miniature adult being dropped into the water ready to go. That, in turn, helps explain why settlement cues matter so much. Researchers have shown experimentally that Osedax japonicus larvae can be kept alive in tanks for about 10 days, and that settlement can be induced by adding pieces of whale bone to the water. MBA has also described the larvae as following chemical cues to the bones of dead animals before settling. That does not mean every natural cue has been completely worked out in molecular detail, and it is worth being honest about that. But the broad picture is clear enough. These larvae are not crashing randomly into the seabed and hoping for the best. They are responding to the presence of appropriate material, and bone itself appears able to trigger the shift from drifting life to attached life. There is something very practical about the first moments after contact. The 2013 developmental study reported that larvae settled on bones, elongated their bodies, and crawled around on the bone surface. Then they secreted mucus to create a tube, and the palps began to develop. That sequence is useful because it turns a vague idea of metamorphosis into something you can actually picture. The larva does not simply touch bone and suddenly become a little adult worm in one magical step. It settles, stretches, explores the surface, starts building the tube, and begins the visible adult structures in order. In other words, once it reaches the right place, the body starts reorganizing quickly around a sedentary life. For years, this stage of the life cycle also seemed to explain the extreme sex difference in a satisfyingly simple way. Earlier work, summarized in the 2015 PLOS 1 paper, proposed that larvae settling on exposed bone develop as females, while larvae landing on females develop as dwarf males. That fit the broad observations very well. Newly settled females on bare bone build tubes, palps, roots, and the ovisac. Larvae that encounter an established female instead can become tiny males living inside her tube. The same study also notes that these leucisthotrophic larvae are the stage that allows dispersal and colonization of new appropriate substrates, so the older model tied settlement site very neatly to later adult role. More recent work has made that story more complicated and better for it. The 2025 EvoDevo study on O. japonicus reports evidence that sex is already determined before metamorphosis, not assigned entirely by the settlement surface. The researchers found that a transient ciliated gut appears in about half of late larvae and identified those gut-bearing larvae as females using sex-specific gene expression. They also found sexually biased gene expression as early as the 2-day larval stage, supporting the conclusion that sexual differentiation happens before the larva settles, so the current picture is more careful. Earlier studies suggested environmental sex determination based on where larvae landed. Newer developmental evidence in at least one species indicates that the larvae may already be male or female before settlement, even though settlement still has a huge effect on what life course becomes possible afterward. That distinction matters because it changes how we imagine the larva drifting through the water. Under the older view, one could picture an almost blank developmental slate waiting for the seafloor to decide its future. Under the newer O. japonicus results, it looks more like the larva may already carry a developmental direction of its own, with settlement deciding whether that direction can be completed in a full female form or diverted into the dwarf male route. Either way, the search itself remains critical. A female larva still needs bone. A male larva still needs access to the right social setting, near or on a female if it is to end up as one of those tiny tube-dwelling adults. The ocean is not making this easy for either of them. Once a female larva has settled on bone, growth can be surprisingly rapid. Earlier developmental work summarized in search results says that female individuals shed their larval traits during metamorphosis and continue organogenesis and extensive growth for 2 to 3 weeks before developing oocytes. The 2013 developmental study then reports that females started to spawn eggs about 6 weeks after settlement. That is a fast turnaround for an animal establishing an entirely new feeding system, picking up symbionts, boring into bone, and becoming reproductively active. The deep sea is slow in many ways, but Osedax does not waste time once the right real estate has been found. It is more like a very determined tenant who signs the lease, renovates the basement, invites in the microbes, and starts a family before the neighbours have finished introducing themselves. The male route is stranger still. The same 2013 developmental study found that recruitment of dwarf males could be induced experimentally by rearing larvae with adult females. Earlier work on Oroseus, summarized in PubMed search results, reported a pattern of initial female settlement followed by gradual male accumulation, and that many females were already fecund about 3 months after deployment on experimental whale bones. This is important because it means the female does not need to arrive already carrying her full future harem. She can settle first, begin feeding, and then accumulate males over time as more larvae arrive. The family builds gradually, one drifting larva at a time. That male accumulation also appears not to come from a single highly inbred household supply. A 2008 study, reflected in later references and search results, concluded that Osedax females and their harems are recruited from a common larval pool. So the dwarf males in one female's tube are not simply all her own sons waiting around forever in a terribly uncomfortable arrangement. They may come from the broader local larval population. That makes the whole system more dynamic and less closed than it first sounds. The female is a settlement site, not just a mother with permanent in-house offspring. A larva drifting through darkness may end up joining a household it has no close family connection to at all. Which is one more reason ordinary words like family have to be used with some caution here. The ability to drift effectively is not just inferred from laboratory work, it is also suggested by distribution patterns in the wild. The 2015 +1 study on Osedax deceptionensis found that specimens from Deception Island and South Georgia Island, about 1,600 kilometers apart, were clearly the same species, and the authors interpreted this as confirming well-developed dispersal capabilities in the genus. That does not tell us exactly how one individual larva travelled, or how often successful long-distance settlement happens, but it does show that Osedax lineages can connect distant sites. The larvae may be tiny, but they are not ecologically trapped in one little corner of the ocean forever. Over generations, they can bridge serious distances. There is an interesting tension in that. On the one hand, the larval stage is short-lived and dependent on yolk. On the other hand, the genus clearly manages broad dispersal. The solution is probably not that each larva crosses impossible distances under its own power like some determined bone-seeking marathon runner. It is that enough larvae are produced, enough currents move them around, and enough suitable substrates appear over time that the lineage maintains connections across regions. Whale falls and other vertebrate remains may act as stepping stones. A larva does not need to find every bone in the ocean. It only needs to find one suitable bone before its reserves are gone. Across millions of larvae and many generations, that can be enough to produce wide geographic spread. Another important point is that settlement is only the start of microbial life for the worm. Because pre-settling larvae are aposymbiotic, the newly settled juvenile must acquire its Oceanospirales partners from the environment. The 2012 BMC study says the worms obtain these essential endosymbionts locally where their larvae settle, and it suggests repeated infections may occur as the roots proliferate through the bone. That means the larva is not just changing shape after settlement, it is entering a new biological relationship almost immediately. The adult feeding system is assembled in layers: first settlement, then metamorphosis, then growth of the root system, then infection by the symbionts, and then continued proliferation of those roots and microbes together. That repeated acquisition model is especially striking because it makes the juvenile feel less like a completed organism, and more like a construction project still taking on new workers. The BMC study found that mixed infections appear to be common rather than exceptional in Osedax individuals, and that no strict association existed between particular host species and dominant symbiont ribospecies in their Monterey Bay time series. The practical meaning is that a juvenile settling onto bone is entering a local microbial market, not opening a sealed kit packed by its mother. The worm must make itself in place, using what the site provides. That is a risky strategy in one sense, but also a flexible one. It lets the lineage respond to the actual microbial environment around each carcass, rather than relying on a one-size-fits-all package from the start. It is also useful to notice how much of this life stage happens without the kinds of cues that matter to us. There is no light to follow. There may be no large structure visible from any meaningful distance. The larva is operating in darkness and on chemistry, current, timing, and contact. Bone cues can trigger settlement in the lab, and MBARI has described the larvae as following chemical cues in nature. But from the larva's point of view, this is still a remarkably difficult search. It must move through water with finite reserves, encounter a suitable vertebrate remains patch, begin metamorphosis promptly, and do all of this without carrying its own feeding bacteria in advance. When people hear zombie worm, they often focus on the adult feeding method. In truth, the larval stage may be the bolder gamble, and yet it works. It works often enough for Osedax to appear in deep and shallow settings, in the Pacific, Atlantic, Southern Ocean, Antarctic waters, and beyond. It works often enough for experimental bones to be colonized in less than a year in some studies. It works well enough that one species can turn up over distances of 1,600 kilometers, and well enough that females can settle, grow, acquire symbionts, become fecund, and begin the cycle again in a matter of weeks after contact with bone. For such a tiny drifting stage, that is a very impressive record. So when we picture Osedax larvae drifting through darkness, we should not imagine helpless specks tossed around aimlessly until one gets lucky. They are better understood as short-lived, yolk-powered dispersers built for one specific job. They leave the female's reproductive system without symbionts, survive for a limited period, respond to the right kind of substrate, and then change very quickly once they find it. Some will become the large root-bearing females fixed into the bone. Others, under the right conditions, will end up as dwarf males inside the tube of an established female. Either way, the entire future of the colony depends on a stage so small that most of the deep sea will never notice it at all. And there is one last reason this drifting stage matters before we move on. A larva that can settle on vertebrate remains, rather than only on fresh whale skeletons in some narrow modern setting, opens a much bigger evolutionary door. It suggests that the search is not necessarily for whales alone, and that the history of Osedax may reach beyond the age of modern whale falls into a wider world of fish bones, reptile bones, and other hard leftovers on the seafloor, which is exactly where the story starts to widen next. By this point, the whale might seem like the whole story. It is large, easy to picture, and impossible to ignore once you know what happens to its skeleton on the seafloor. But Osedax is bigger than whales in one very important sense. Whales are where scientists first found these worms, and whale falls remain the clearest stage on which they perform. But the evidence now shows that they are not simply whale specialists waiting around for modern cetaceans to die. ARMB's current overview says various Osedax species can colonize a broad array of bones from fish, marine mammals, birds, turtles, and terrestrial mammals, and it places the genus across depths from 10 to 4,000 meters. That is already a much wider menu than the original discovery might have suggested. That broader diet changes the whole evolutionary picture. If Osedax can use many kinds of vertebrate remains, then its history does not need to begin with whales, and it probably does not. Scientists were pushed toward that conclusion surprisingly early. In 2008, a Royal Society paper showed that Osedax colonized cow bones deployed in Monterey Bay at depths from 385 to 2,893 meters. Colonization happened as quickly as 2 months after deployment. Some females on the cow bones were already producing eggs, and some hosted dwarf males in their tubes. That result was important because it broke the old idea that these worms were tied only to whale falls. If they can settle, grow, and reproduce on cow bones lowered by researchers, then whales are not their only acceptable meal. They are simply one very large and convenient one. There is something almost funny about that experiment once you picture it plainly. Scientists had already found a worm that eats whalebone in the deep sea. The natural next question was whether it was truly a whale specialist. So they lowered cow bones into the ocean and waited to see what would happen. What happened was that the worm showed up and got to work. It is one of those moments in science where the method sounds mildly ridiculous, until you remember that it answered a serious question very neatly. The result forced biologists to stop thinking in terms of whale worm, and start thinking in terms of vertebrate hard parts that can persist long enough and contain enough usable organics. That is less catchy, admittedly, but a lot closer to the truth. Once that broader feeding ability was established, other lines of evidence started to line up behind it. MBARI now states directly that various Osedax species colonize bones from fish, birds, turtles, terrestrial mammals, and marine mammals. A 2023 paper on shark teeth, discussing the genus more broadly, says extant Osedax have been shown to live off exposed bones of a range of vertebrates, including teleost fish. So the pattern is no longer a speculative extension from one or two odd experiments. It is the mainstream view of the genus. Osedax is a specialist, yes, but it is a specialist on a type of resource rather than on one particular charismatic host. That distinction matters because it helps explain why the worms could have a much deeper past than whales. Whales, in anything like their modern forms, are geologically recent compared with the long history of vertebrates in the oceans. If Osedax depended strictly on whale bones, then the lineage would have to be quite young. But if it can use fish, birds, reptiles, turtles, and terrestrial mammal bones that happen to reach the sea, then the door opens much wider. Fossil evidence has steadily pushed that door backward in time. In 2010, researchers reported fossil traces of Osedax in early Oligocene whale bones from western Washington. In 2011, another study reported borings in fossil marine bird bones and concluded that Osedax was, and most likely still is, able to consume non-mammalian bones. In 2013, further work documented traces in Oligocene whale teeth and fish bones from the same broad region. That is already a strong sign that the worms had a more varied diet tens of millions of years ago than the original whalefall discovery alone would have implied. The bird bone result is especially useful because it interrupts the tidy assumption that the worms must have evolved around marine mammals and then only later broadened their taste. The PubMed summary for the marine bird paper says the fossil borings were found in bones of early Oligocene flightless diving birds of the family Plotopteridae, and that the worms responsible lived at densities of up to 40 individuals per square centimetre. That is not an accidental nibble on a strange substrate. That is successful colonisation in numbers. A bird bone on the seafloor is not as grand as a whale vertebra, but from Osedax's point of view it may still be a workable patch of food if it lasts long enough and contains the right kind of material. The fishbone evidence pushes in the same direction. The 2013 Paläontologische Zeitschrift paper reports traces of Osedax in Oligocene whale teeth and fish bones from western Washington. Search results for that paper note that the boreholes were investigated with microcomputed tomography and that the fossils came from deep water strata. This matters because it shows the trace morphology was not just vaguely suggestive. Researchers were comparing the internal forms of the borings with those made by living Osedax and treating the resemblance as strong enough to extend the known record of the genus's habits. Fish bones are a very useful test case here because they point away from a world dominated by giant whale carcasses and toward a more general marine scavenging niche on vertebrate remains. Even more intriguing is what has happened with teeth. In fossil material, researchers have reported traces in Oligocene whale teeth. In Living Systems, a 2023 Cambridge paper documented Osedax using shark teeth as a food source. In that study, jaws of the common thresher shark were deployed on the seafloor at around 1,000 metres depth off Monterey. After 8 months, 2 Osedax species were found boring into the roots of some of the teeth and using the dentin pulp, which is rich in collagen, as food. The enameloid crowns and the tessellated cartilage showed no signs of Osedax activity. That result is especially interesting because the authors call it the first direct evidence of Osedax exploiting a food source that is not bone. Strictly speaking, the worms were still going after a collagen-rich hard tissue, but not one that most people would casually file under bone at all. That shark tooth result does 2 useful things at once. First, it shows that Osedax is interested in collagen-rich internal tissues rather than mineralized hard parts in some abstract sense. The worms did not eat the hard enameloid crown. They targeted the dentin-rich root and pulp region instead. Second, it raises a larger evolutionary question that the authors spell out directly. If Osedax can use dentin in shark teeth, then perhaps the original food source of the lineage was not bone in the narrowest sense at all, but a broader class of collagen-rich vertebrate hard tissues available on the seafloor. The paper explicitly says this finding raises questions about the original food source of Osedax bone worms and suggests examining fossil sharks, bony fish and reptiles for traces. It is rare for a single weird result to make the old name of an animal feel slightly too narrow, but this one does. Of course, not every hard vertebrate tissue is equally attractive or equally accessible. The same shark tooth study found no evidence that Osedax used the enameloid crown, and no evidence that it exploited the tessellated cartilage fragments that remained from the jaws. That is a useful reminder that beyond whales does not mean anything hard from any vertebrate will do. The worms are still constrained by chemistry and structure. They seem to target the parts rich in collagen and other usable organics, and to avoid surfaces that are too hard, too poor or too sealed off to be worth the effort. So the broader diet is real, but it is not careless. Osedax is adventurous in the way a specialist can be adventurous, by recognizing the same kind of usable material inside very different leftovers. The fossil record then pushes the timeline back further still. In 2015, a biology letters paper reported fossil traces of Osedax in Cretaceous marine reptile bones, arguing that the worms lived on Mesozoic marine reptile deadfalls. More recently, a 2024 proceedings of the Royal Society bee paper reported new occurrences from late Cretaceous marine reptiles, including plesiosaurs and, for the first time, mosasaurs, extending the known Cretaceous record across both sides of the northern Atlantic Ocean. That is a major shift in perspective. It means Osedax was likely exploiting marine vertebrate remains long before the great radiation of modern whales filled the oceans with whale falls. The lineage may have passed from one set of giant vertebrate carcasses to another across mass extinctions and major faunal turnovers, staying with the resource rather than with any one group of animals. That is one of the most satisfying things about this entire subject. At first, the worms seem almost absurdly tied to whales. Then the evidence keeps widening the frame. Cow bones in Monterey Bay, fossil bird bones, fish bones, whale teeth, shark teeth, late Cretaceous marine reptiles. Each new piece makes the older picture look too narrow. By now, the safest statement is not that Osedax evolved to eat whale bones, but that it evolved to exploit vertebrate hard tissues on the seafloor wherever and whenever suitable ones appeared. Whales are simply the most obvious modern expression of that opportunity, because they are so large, so lipid-rich, and so durable once they reach the bottom. There is another useful consequence of that broader view. It helps explain why the genus could diversify so successfully. Embury says 32 species have been discovered worldwide so far, and the worms occur from shallow settings around 10 metres down to about 4,000 metres. A lineage tied only to rare deep whale carcasses might still survive, but it would have a harder time building that much diversity. A lineage able to use a wider array of vertebrate remains across different depths and regions has many more chances. Different carcass sizes, different bone densities, different local scavenger communities, different currents, different sediment conditions, and different microbial environments all provide opportunities for divergence. The resource is specialised, but it is not vanishingly singular. Even so, it is important not to oversimplify in the other direction. Beyond whales does not mean that small fish bones on the seafloor are equivalent to whale falls in ecological importance. They are not. A whale fall is still extraordinary, because it supplies a huge pulse of organic material and can support entire successional communities for years or decades. A fish skeleton or isolated bird bone is a much smaller and more fleeting opportunity. The point is not that every hidden meal is equally grand. The point is that from the worm's perspective, a smaller meal can still count if it persists long enough for settlement and growth. This is one reason fossil traces matter so much. They show that Osedax could make use of remains we would never have guessed from the original discovery alone. The fossil trace work also gives paleontologists something very useful, a way to infer ancient ecological interactions from the holes themselves. Osedax borings have characteristic forms, and researchers have used CT scanning and comparisons with modern worms to recognise them in ancient bones. The 2024 Proceedings B paper emphasises that recognition of Osedax in the fossil record depends on unique features of the borings defined by the organism's morphology, and the 2014 paper on boring diversity notes that experimental and fossil borings in fish bones resemble those made by living species. That means the worm is not only interesting as a living deep-sea animal, it is also a maker of trace fossils, leaving a record of meals taken millions of years before anyone knew the animal existed. There is something quietly impressive about that. The animal itself is soft, small, and unlikely to fossilise well. But the damage it does to bone persists. The worm disappears and the meal record remains. Paleontologists then recover a fossil rib, tooth, bird bone or reptile element and see the work of an animal that, in life, spent its time hidden in darkness beneath the seafloor. It is a good reminder that some lineages leave history not by preserving their bodies, but by carving their habits into the bodies of others. Osedax is not famous because its own body fossils fill museum drawers. It is famous because it writes in bone. The newer work on dense whale ear bones adds one more nice twist. A 2025 paper reported fossil traces of Osedax in an Oligocene whale tympanic bulla, which had earlier been thought too dense and nutrient-poor to attract or sustain the worms. That matters because it suggests Osedax may be willing to exploit even some hard tissues previously considered poor candidates, provided the right entry point and enough usable organic matrix exist. It is not proof that the worms will eat everything with collagen forever, but it does show that their opportunities may continue to surprise researchers. Every time someone thinks a certain bone is too dense, too odd, or too specialised to be worth the effort, Osedax seems tempted to have another look, and perhaps that is the most useful lesson of this entire section. The other hidden meals are hidden partly because they are smaller, older, or less obvious than a whale skeleton resting on the seafloor, a fish bone in ancient sediment, a bird bone from the Oligocene, the root of a shark tooth, a marine reptile vertebra from the late Cretaceous, even a dense whale ear bone. None of those announces itself with the same dramatic force as a whole whale fall, but taken together, they reveal a lineage far older and more flexible than the original discovery suggested. Osedax is not merely a specialist of modern whale death. It is a long-running deep-sea opportunist of vertebrate remains, following collagen-rich hard tissues across hosts, habitats, and geological eras. That broader history makes the worms feel less like a biological curiosity and more like a durable solution to a recurring problem. Big vertebrates die, their remains sink or are buried. Hard tissues rich in useful organics persist after softer tissues are gone. Most animals cannot exploit those leftovers efficiently. Osedax can. Whether the host is a whale, a bird, a fish, a turtle, a terrestrial mammal dropped into the sea, or perhaps even the root of a shark tooth, the same basic opportunity keeps appearing in different forms. The lineage does not need one kind of giant host to survive. It needs vertebrate remains that last long enough and contain enough usable material. That is a much older and more stable niche than wait for a whale. And once you see that, the closing part of the story changes as well. These worms are no longer just one more deep-sea oddity living on a spectacular carcass. They become part of a larger truth about the ocean floor, which is that death there is not a clean stop but a transfer, a long and very practical redistribution of locked-up material into new lives. Osedax happens to perform one of the strangest jobs in that transfer, and perhaps one of the most revealing, which is where the last part naturally needs to go next, away from the menu, and toward what the whole arrangement says about death, renewal, and the way the deep sea refuses to waste anything useful. What all of this adds up to is surprisingly plain once the shock of the details wears off. A whale dies, its body sinks. Large scavengers strip the soft tissue in months. Organic scraps enrich the nearby sediment for more than a year. Then the bones continue feeding life for years or even decades, in part because they still contain useful lipids and proteins. In that long later stage, Osedax becomes one of the animals that decides what happens next. Noah describes whale falls as a sudden, concentrated food source in the deep sea, and MBARI notes that Osedax can remove visible evidence of a whale skeleton in as little as a decade. That is a strong reminder that the skeleton is not the end of the food story. It is simply the point where the menu changes and the specialists take over. That matters because bones look permanent to us. They look like the part built to last. On land, they often are. In museums, they last even longer. In the deep sea, though, durability and safety are not the same thing. A whale skeleton may sit intact for a while, but if Osedax arrives, the bone is no longer just hard structure. It becomes habitat, food source, and work site. Smithsonian explains that these worms do not eat mineral bone directly. They secrete acid that dissolves the bone enough to free trapped fats and proteins, and then symbiotic bacteria in the worm's body help digest those nutrients. That feeding system turns something that looks sealed and final into something open and useful again. That is one of the clearest things bone-eating worms reveal about death. Death in the deep sea is not neat. It is not a clean stop followed by stillness. It is a redistribution problem. The soft tissue goes first because it is easy to reach. Smaller scavengers and sediment dwellers take advantage of the leftovers next. Then the bone itself becomes available to animals and microbes that can work on harder material. NOAA's overview of whale falls describes exactly that succession, from scavengers to enriched sediment communities to later stages supported by the skeleton. Once those steps are laid out plainly, Osedax no longer looks like a grotesque exception. It looks like one logical answer in a long chain of consumers, each using what the last stage left behind. It also tells us something practical about renewal. Renewal here does not mean anything sentimental. The whale does not come back. The bones do not heal. Nothing is repaired in the ordinary sense. What happens instead is transfer. Carbon, proteins, lipids, and other materials locked inside a large body move outward into many smaller bodies over time. MBAR says Osedax plays a major role in controlling the fate of whale bones, while Noah describes whale falls as new food webs forming around one large carcass. So renewal in this setting means that material moves on. It leaves one large animal and enters worms, microbes, crustaceans, fish, sediment communities, and later generations of larvae. It is less like recovery and more like thorough reuse. That transfer becomes even more interesting when you remember how specialised the worms are. Osedax females have no mouth and no stomach, yet they feed efficiently by rooting into bone and working with microbes beneath their skin. Smithsonian says the worms lack a mouth and stomach and rely on acid plus bacteria to free and digest fats and proteins inside the skeleton. The 2023 Nature Communication study adds that the Osedax oceanospiralalis symbiosis has genomic adaptations for bone digestion, including host changes tied to amino acids abundant in vertebrate bone and symbiont pathways that can help handle bone-derived lipids. So this is not a crude or temporary workaround. It is a deeply integrated way of life, built around the fact that dead vertebrate hard parts still contain a lot of food, if something can get to it. That same specialisation also says something about the deep sea itself. People often picture the abyss as empty because it is dark, cold, and out of sight. In reality, it is full of organisms waiting for the right kind of resource to appear. NOAA describes whale falls as a bonanza in the deep sea. The Frontiers Review found by search describes them as oases in food-poor surroundings. Those words fit because the surrounding seafloor usually runs on small scattered inputs. A whale fall changes the local balance so dramatically that entire communities can gather and persist. Osedax is part of that larger truth. It exists because the deep sea is not passive. It is prepared. The animals, microbes and larvae are already there, and when a suitable carcass arrives, they respond. The worms also reveal that unusual body plans do not appear for entertainment value. Nature is not trying to impress us with weirdness. Osedax looks bizarre because the job is unusual. A worm living on a rare, hard, nutrient-rich substrate in darkness does not benefit much from eyes. It does not need a normal gut if the food must be dissolved and processed at the surface first. It does not need large free-living males if habitat is patchy and mates are hard to find. MBARI's project page summarises that combination very well. The genus includes species that colonise many kinds of vertebrate remains. Most species have dwarf males living in female tubes, and the worms can reduce a whale skeleton quickly once established. Every part of that arrangement fits the same general rule. Nothing extra is kept unless it helps on the bone. Another important point is that Osedax is not just about whales. MBARI says species in the genus colonise bones from fish, birds, turtles, marine mammals, terrestrial mammals, and more. The Royal Society paper on cow bones showed that the worms could settle, grow, and reproduce on non-whale vertebrate remains in Monterey Bay. That wider menu changes the meaning of the whole lineage. It suggests that Osedax is not a recent specialist invented by modern whalefalls alone. It is better understood as a durable solution to a recurring problem: how to exploit vertebrate hard tissues after softer parts are gone. Once that becomes the focus, the worms stop looking like creatures tied to one dramatic event and start looking like long-term users of hidden meals wherever those meals last long enough. That broader diet is one reason the evolutionary story is so strong. Mbari says that since 2004, scientists worldwide have discovered 32 species of Osedax from depths of about 10 to 4,000 metres. That is a lot of diversity for an animal first noticed on one grey whale skeleton in 2002. It means the strategy works in many places and under many conditions. It also means that vertebrate remains on the seafloor, whether large or small, must be common enough over long stretches of time to reward this kind of specialisation. The worms did not become this diverse by waiting for one rare lucky meal. They became diverse because the ocean has kept providing versions of the same opportunity again and again in enough places for different lineages to persist and diverge. There is also a useful lesson here about scale. When people hear about bone-eating worms, they often focus on the small animal because it sounds dramatic, but the better way to look at it is from the scale of the whole carcass and the whole ecosystem around it. NOAA says different stages of whale decomposition support different biological communities, and the whalefall can keep feeding deep-sea life long after the obvious flesh is gone. Osedax belongs to that larger schedule. It does not arrive to dominate the entire story from the first minute. It comes in when the conditions suit it, takes over a difficult part of the job, and in doing so changes what happens to the last major store of usable material. That is not theatrical, it is efficient. And efficiency is usually what matters most where food is scarce. The bacterial partnership beneath the worm's skin makes the same point in another way. The 2023 Nature Communications paper says the host and symbiont show complementary genomic adaptations for bone digestion, and Smithsonian says the worm would not manage this diet without symbiotic bacteria helping digest collagen, cholesterol and lipids. So even at the level of one individual animal, the lesson is the same as at the level of the whole whalefall. Nothing useful is left idle if some combination of organisms can get at it. The worm does not do the whole job alone. The bacteria do not do the whole job alone. Together they use a resource that many other animals would leave behind. That is a very common pattern in biology, just made unusually visible here because the food happens to be a whale skeleton on the seafloor. There is a second lesson in that, and it is less about death than about limits. Even a whale, one of the largest animals on Earth, becomes local once it reaches the seafloor. A huge body turns into a fixed patch of food. A worm only a few centimetres long can then help decide how quickly that patch disappears. MBAR notes that Osedax can erase visible evidence of a whale skeleton in as little as 10 years. That is a useful correction to the way size often shapes our thinking. Large animals feel permanent while they are alive. On the deep sea floor, their remains become part of the same practical cycle as everything else. Size changes the length of the process, but not the rule. If something contains useful nutrients, something else will eventually use them, and that may be the calmest thing this subject has to offer, not because it is soft or comforting in the ordinary sense. It is not. A bone-eating worm is still a bone-eating worm, and one should not pretend otherwise. But there is a steadiness to the system. The deep sea does not waste a whale. It does not hurry either. It just continues. The carcass becomes food. The bones become food. The food becomes worms, microbes, larvae, scavengers, and enriched sediment. Then those lives support other lives in turn. NOAA's Whalefall Overview and MBAR is long work on Osedax. Both point back to the same basic fact. What looks like an ending from one angle is, in ecological terms, a long sequence of uses. So when we leave these worms where they belong, down on the ocean floor among the last remains of a great skeleton, the main thing they leave behind is not just surprise, it is perspective. They show that the deep sea runs on patience, on specialisation, and on the steady transfer of material from one body to many others. They show that hard leftovers are still food. They show that even a soft worm with no eyes, no mouth and no stomach can become one of the most important late-stage consumers in the life of a whalefall. And they show very clearly that far below the surface, death is followed not by emptiness, but by work. Sleep well, and let that last image stay simple. A dark seafloor, a white skeleton, and small red worms carrying on with a job the ocean has had ready for them all along.